Thanks, Adam. Good morning. It's 9 o'clock, so we're going to go ahead and get started. Welcome to day 2 of our FDA M-CERSI workshop on accelerating product development in pediatric SLE. We had a great discussion yesterday. We had approximately 190 participants online. So we're looking forward to the discussions today. We're going to pivot a little bit today. Session 4 is going to focus on safety assessment and trial design considerations. And I'm going to invite the first speaker for today, Mary Beth Sun, to provide an overview of safety assessment in pediatric SLE, challenges and opportunities.
Hi, good morning, everyone. I want to thank the organizers for putting this very important workshop together and for the opportunity to talk with you this morning. These are my disclosures. So briefly, I'm going to go over pediatric-specific safety considerations. What do we actually know about safety data from pediatric lupus, and what are the challenges of capturing rare events, and what strategies can we use? So what are the safety concerns in pediatric lupus? So people talk a lot about short-term versus long-term safety events, and I have to say I've been struck by some of the conversations about like what I think is long-term versus what the other parameters are for long-term. So we take care of children and adolescents. We think about them now and also 20 to 30 years from now. So I think there's different horizons for what we call long-term. Short-term is much easier. Things like infections, hypersensitivity reactions to infusions, side effects, tolerability, which impacts adherence, and then impact on growth while the child is still growing are all important considerations. And then long-term things like growth and bone health, puberty and future reproductive health, central nervous development and what the impact that is on adults who've had pediatric onset disease, and then risk of malignancy. And it's important to state that the risks of these are governed by the specific properties and the mechanism of action of the medication in question, and it's going to vary across medications. So is safety different in children? This is one of the key questions to think about. So children are actually healthier in many ways than adults. So they have fewer baseline risks for infection and comorbidities. We see this every day in clinic where patients just get fewer infections on the pediatric side. Children have fewer concomitant medications because they're just healthier. But importantly, they are still developing physically, neurologically, and emotionally. So what do we think about in terms of what are important safety considerations in children? So children grow, so their bone health is an important issue to think about, and the majority of bone is deposited in adolescence. And peak bone mass is achieved somewhere between 20 to 25 years of age. And so if we are giving medications that lead to perturbations in peak bone mass, that's something that they're going to live with for the rest of their life. And just to say the obvious thing here, we're talking about glucocorticoids here, right, that impact bone health the most significantly. And in fact, there's studies in adults to say that cytokine blockers like IL-6 blockers and TNF inhibitors actually protect your bone mass. This is specific to pediatrics. We have to think about a final height attainment as well as puberty. Oh, good. Okay, so what I'm showing over here, the graph on the left, this is the impact of disease onset of pediatric lupus, premenarchal versus postmarchal. And what you see, the bar on the left is final height attainment for children who developed lupus before menarche. These are about 400 female patients from Canada. The bar on the left is final height attainment for those who developed lupus after menarche, and the difference there is 3 centimeters. So that's a permanent impact on their growth. Another important aspect of growth, this is an international cohort. The total cohort is about 550 patients with lupus. They separated out the females, and what you're looking at here in the red box is that there's actually delayed or absent menarche in about 20% of patients, which also has ramifications for bone health and other things. And again, just to stress what we all know, glucocorticoids here are the main culprit in impacting both final height attainment as well as impact on puberty. We get this question a lot in clinic, what's going to happen to my child in terms of having children later in life? We talk a lot about cyclophosphamide. So as we know, there's increased risk of ovarian failure. This is dose dependent, and it also depends on when you get the cyclophosphamide. But just to point out that if you give a 15-year-old a course of Cytoxan, that might be the first of a few cycles of Cytoxan. And so risks of malignancy and infertility increase with each round. So there's efforts to use like the lower dose, your lupus protocol, but still an important aspect of taking care of these patients. And then lastly, malignancy risk. What do we know about the malignancy risk in pediatric lupus? So there was one study that utilized 12 lupus registries in North America, and they linked them to their regional cancer registries. And this was actually about 1,200 patients going all the way back to 1974 through 2009. The mean duration of follow-up was about 7.6 years, and they calculated standardized incidence ratios. And it looks as though there is a higher incidence of all cancers with the SIR of about 4.35. The CIs are a little wide. And non-Hodgkin's lymphoma was the most common. Do want to stress these were rare events. Okay, it's about 1.6 malignancies per 1,000 patient years. But nonetheless, this is a signal that we should continue to follow. And how does this compare to adult lupus studies? And there's another study in adult lupus patients where the risk of non-Hodgkin's lymphoma was also about 4.3. Just to point out, when they subsetted that cohort of adults, it was actually the younger patients were at higher risk for malignancy. And that's probably because they had pediatric onset disease and had years of immunosuppression. So what this all means is that it's actually malignancy is uncommon, but risk may be increased relative to the general pediatric population. And this is something that we should be thinking about in terms of long-term safety. Okay, and then of course, we've talked a lot about developing brains. Anyone who's been around an adolescent is well aware their brains are still developing. So what really happens at the level of the brain? So gray matter peaks around 10 to 12 years. Frontal lobes, where all your complex thinking happens, does not get fully developed until you're in their 20s. And one thing, just to point out, those children who have neuropsychiatric lupus, as it turns out, your prefrontal cortex, which is responsible for complex cognitive function, is most vulnerable at ages 14 to 16. And that is one of the ages that's peak onset for childhood lupus. And so you also have to think about the impact of the disease on the developing brain. So how do we put this all together? What are the risks and benefits of immunosuppression in pediatric rheumatic disease? So certainly we think a lot about infection risk. Severe infections have a negative impact on growth and development as well. There's possible neurodevelopmental effects. There's impact on growth, and there's maybe an increased risk of malignancy, all based on medication use. But the benefits of aggressively treating pediatric lupus outweigh these risks because you control the inflammation, decreases end organ damage, improves outcomes, and importantly, the more steroid-sparing options we have, we decrease glucocorticoid exposure. And we know that children with pediatric lupus get more steroids than adults with lupus. But just to say, this is a challenge. This is going to be challenging to do well. So I'm just going to take the example of impact of having lupus as a global term, the net effect of growth and development. There's multiple things that impact your growth and development. So on the left, you have disease activity with deleterious effects on growth and development. So there's inflammation, there's poor nutrition, delayed puberty, impaired growth. Steroids do have all the effects we've talked about: growth suppression, bone toxicity, metabolic effects. But on the other side, non-glucocorticoid-based treatments facilitate normal growth and development because they treat they control the inflammation, they are steroid sparing, and they provide immunomodulation. So disentangling all these different impacts on all these different things on the final outcome of growth and development is going to be challenging. So what do we have? So we've talked a lot about the Pluto trial. Here I just pulled out specifically the safety data that was provided by the Pluto trial. Safety events were similar between the treatment group and placebo group. Serious adverse events were also similar between the two groups, and the one death that occurred in the trial was in the placebo group. This is another helpful study. This is pooling of the data from the adult studies, all the BLISS studies, plus EMBRACE with Pluto. And what you can see, what you're looking at here, oh, sorry. Okay, sorry. This table down here lists all of the adverse events that happened across the childhood, the Pluto trial, and the adult studies. And what you can see is that the adverse events, including serious adverse events, were not significantly different between these trials. And then, in terms of like other data that we have, so just to state the obvious, we don't have enough. The top two boxes, so the first one is looking at rituximab use. This is a pretty large single center study. This was rituximab used for any indication, just to be clear, because they wanted to pool as many patients as possible. There were very few adverse events. Infections were an issue. It's not an unexpected result. The middle box refers to a recent meta-analysis. They pooled 14 studies. And what they actually found is that SAEs were infrequent, and in fact, there were fewer adverse events and fewer infections, fewer risk of flares in the treatment group as compared to the groups that did not get the belimumab. And then again, just to emphasize, there are data proving that use of glucocorticoids in pediatric lupus leads to toxicities. So here, the authors applied the GTI, the glucocorticoid toxicity index, to a group of kids with lupus, and there were clear signals for BMI, blood pressure, mood, and sleep. So these are all impacts directly from glucocorticoids. Okay, so do we have what we need? I think people in this room can agree we do not have everything that we need yet for safety data. That's why we're here. Short-term safety, from what we know so far, is likely similar, and the adult data could be leveraged. But I don't think there's still a lot of work to be done, because what patients and families want to know about is long-term safety of these medications for their children and loved ones. So things like final height attainment, serious infections, infertility, malignancy, this is the long horizon, okay? And so this is going to take some work. Looking at this is also complicated by disease severity, because there's confounding there. You get more medicines the sicker that you are, so there's confounding by indication. We tend to use a lot of medications, and so there's difficulty figuring out which medication did what. These are rare events, and the risk window really is unknown, but probably long. So where do we start? What do we think that we need? So we definitely need systematic collection. Missing data here might occur in the highest risk groups, and we'll miss the signal. So it needs to be systematic. It needs to be longitudinal, because these are rare outcomes, so you need long-term data to identify signals. I think we know most of the outcomes that we care the most about, so we have multiple outcomes to follow. And as I said, we really need to be able to attract medication combinations over time to see if we can elucidate which one is causing the issue. And I just want to say that many of these features are age independent. The adult lupus patients care about these things as well. So are there ways that we could synergize? However, I'm going to throw in a little quid pro quo. I still think pediatric populations are different in some important ways, which just means that we need to track this. So as we all said, we know they get more steroids. So what does that mean for them in terms of all the outcomes that we think about? Okay, so they have a higher glucocorticoid burden. This is because there's not enough medications approved for pediatric lupus. As Dr. Sadoun pointed out, there's a higher genetic burden. There could be a higher malignancy risk. And because if you start following them at 15, we probably have to follow them until like they're 40 or 50 for us to know. So it's a long-term follow-up that we need. Okay, so I definitely don't have the answer to how we do this exactly right. Here are some suggestions. I think this is one of the things we should discuss in the panel. So what do we do? What do we do? How do we figure this out? So phase 4 studies are a well-established post-marketing tool to follow patients to see what kind of signals come up. The FDA does this a lot. We have some good data. There are registries with medication and long-term follow-up data. So the EMA 2 years ago put together a real-world data repository of studies. And so we have that as well. And the long-term follow-up data there can be very helpful. We have administrative databases, right? So could we link a database of children with lupus with the the SEER index, which follows malignancies? Could we link it with the kidney transplant database? Could we somehow meld these tools that we currently have, which are imperfect, but could we get some data from there? And then the question arises that as these trials for children and adolescents with lupus roll out, could we register them upon entering the trial for long-term follow-up? And I just want to mention, so the CARE registry, this is one of the goals in addition to phase 4 studies. We really wanted to follow these kids and young adults long-term, even after leaving pediatric care, to collect both safety events, all serious adverse events, as well as long-term follow-up with some PROs that are collected routinely from those patients. So there are ways of doing this, but I just worry that without a systematic way of doing this, that we could miss some of the important signals. So in summary, there's some reassuring data for short-term safety outcomes. Long-term safety outcomes are rare, so we need to study as many children as possible for as long as possible, and collaboration amongst all stakeholders is going to be needed to optimize this kind of data collection. And I thank you for your time.
Thank you. That was great, Mary Beth. Thank you for providing the academic perspective. We now have Laurie Conklin from J&J to provide the industry perspective on safety assessment and extrapolation.
Thank you, Lily, and thank you, Dr. Sun. I was asked to give this talk on extrapolation of safety from an industry perspective, which is a little daunting. I'm a pediatrician, pediatric gastroenterologist, but I've worked within the FDA as well as within academic clinical practice, and I've been in industry for the last 10 years in both small pharma and larger pharma. These are my disclosures. So I'm going to start by saying first that there are obvious tensions between wanting more safety data pre-approval and wanting faster access to new therapies in pediatric lupus. Our typical approach is that we're negotiating plans with both regulatory agencies. We historically have been generating randomized controlled data in pediatric lupus. During that time and other diseases as well, there's off-label prescribing or lack of access to therapies in some parts of the world while awaiting the pediatric data. And that leads to the intended outcome, which is a stronger label and more certainty around the safety in pediatrics. But there are problems with that approach. Our randomized controlled trials are generally unappealing, as we've been discussing over the last 2 days, due to risks associated with placebo. And even things like sham injections, it's a lot of burden on patients, as we've heard from Dr. Brunner as well. Diminishing equipoise over time, so there becomes less and less uncertainty in the pediatric community about the therapies, and less motivation and inclination to enroll your patients in a clinical trial. In the end, we generate relatively small pediatric data sets. And so in comparison to the much larger adult safety data sets, we're generating these small data sets. It's hard to draw conclusions from them. This is a real difference from pediatric rare diseases where you don't have an adult data set to draw any inferences from, and you're really starting from scratch assessing your pediatric safety. And then in that time, which is often years, where physicians are using the drug off-label, they don't have information on dose. And so the clinical practice can develop in different ways where the safety data isn't robust and difficult to interpret. So can and should extrapolation of safety streamline development in pediatric lupus? The old mantra was that safety cannot be extrapolated. But the 2023 ICH E11A guidance really changed that. And it doesn't include pediatric extrapolation of safety in that guidance document. And a high-level view is that the extrapolation guidance talks about assessing similarity of disease, drug pharmacology, and response to treatment in your reference population to your target population. And that target population could be different segments of the pediatric age range. According to the guidance, would develop an extrapolation concept, which integrates all available evidence, evaluate the strength of that evidence, identify uncertainties and knowledge gaps, and then create your extrapolation plan, which is essentially your plan for creating data to fill those gaps, and then generate data according to that plan. And this is important, submit the data in support of a product benefit risk assessment in pediatrics for your pediatric indication, so that it fits within the benefit risk assessment in that indication and in those populations, target populations. So according to the guidance, what the agencies, both agencies, because this is an International Council for Harmonization document, they are expecting from industry an assessment, an evidence portfolio that supports the extrapolation approach. That includes non-clinical data, so information about mechanism of action, ADME, for example, non-clinical pharm tox, information about on-target effects and off-target binding. Clinical data, that might include other populations, other indications, or if it could be that you've already generated data in adolescence and you're using those clinical data in support of your extrapolation concept to younger subsets. And then other existing knowledge. The guidance specifically refers to real-world evidence and literature data as being fair game for inclusion in your extrapolation concept. And you're asking the question in generating your plan, do these data predict a risk of a divergent or a unique pediatric safety risk in those target age subsets? And where there's a low likelihood of a pediatric divergent or a pediatric unique safety concern, development should be streamlined with an extrapolation plan. And that's intended to avoid enrolling children in unnecessary studies. And so in such a case, there could be tailored safety generation. And again, this is just hypothetical, but maybe it's an open label safety study. Maybe it's data supporting your formulation or device that is going to be used in that pediatric population. Maybe it's post-approval monitoring, but it should be rational and it should be supported by data in that extrapolation concept. And then there are circumstances where there is a higher risk of pediatric divergent safety concerns. And in that case, the sponsor needs to assess the benefit risk in those populations and make a decision either to appeal for a PSP and PIP waiver to not pursue study in pediatrics because that risk is too significant, or to design study to answer those safety concerns. And so, that is part of the benefit risk discussion that happens in this extrapolation concept. So I'm going to go through a few things that impact how we would make a decision that there's the potential for pediatric divergent safety risks. One of these is age-specific biology. We've talked about this already yesterday. Infants have a much higher risk plausibility because of the ontogeny, those developmental changes that are affecting PK in infants. There's also significant maturity that's occurring in infancy. And so the safety profile, the safety concerns would be different in infants. When you're looking on the other age spectrum in adolescence, in most cases, you're using the same formulation, the same presentation, the dose is very likely to be the same, and you're really focusing in on what differentiates an adolescent from an adult, and where in those areas might there be a pediatric specific safety concern, like puberty and growth or neurocognitive development. One also needs to consider the type of drug that you're dealing with, whether it's a large molecule, monoclonal antibody, or a small molecule, the components of that drug, whether it's a combination therapy, whether there's special modifications to the therapy that might have a differential effect in a child. And again, a lot of these concerns are in infants. So for small molecules, you've got that ontogeny aspect, that developmental aspect. Difference in concomitant medications used in children should be considered. We've heard yesterday that the disease, pediatric lupus and adult lupus patients generally receive the same types of medication in their treatments, but that's something also to consider in your extrapolation concept and plan. And then this is important and this article was published recently by Gil Burkhart out of FDA in that the FDA wants to hear about on or off-target effects that are potentially relevant to pediatrics. So how might you do that? Well, there are panels that assess secondary pharmacology and off-target binding, and we ought to be considering whether those targets play a role in developmental biology. For example, if you had a drug that had off-target binding at estrogen receptors or modulated in some way the growth hormone IGF-1 pathway, then there would be a higher likelihood that your drug could have safety concerns in pediatrics, and you would need to consider that and factor that into your extrapolation plan. But if not, then it reduces the likelihood that there would be a pediatric divergent safety concern. We've also talked about, it's important to realize that when we talk about this large adult data set from which we're drawing conclusions, we have this large safety data set in adults at a certain exposure range. Beyond that exposure range, we don't have data to support the safety. And so our extrapolation approach generally targets that safety, that exposure range that has been shown to be safe and effective in adults. So our pediatric extrapolation plan might incorporate confirmation of expected exposures. We're modeling the dose and then we're confirming the exposure in pediatrics. Sometimes quantitative systems pharmacology modeling can indicate an expected toxicity or lack thereof, and that could be included within the extrapolation concept to reduce the likelihood of a pediatric-specific divergent safety concern. And it's important to realize that with SLE, we're not dealing with those, except for monogenic lupus, of course, but we're generally studying down to age 5 for many of our programs where we're not getting into the toddler infant risk plausibility. Because I'm from industry and I'm talking about challenges that are related to safety extrapolation in industry, I thought I'd just put in some general high-level industry perspectives. We are dealing with a fast-moving, high-risk adult program. We're often not, companies aren't necessarily wanting to make big investments before they know that the phase 2 is successful. We see that RCTs enroll slowly, particularly with placebo or active comparator arms, that they may have failed already, so that standard of care that they've already failed. So it's daunting to be setting out onto a study where you know there's going to be significant challenges with enrollment. Long-term safety registries, I'm talking 10 to 20 years, they are often quite challenging operationally for the reasons that Dr. Sun had described with confounders, and also with, we see this in other diseases as well, where practice shifts over time. So people start using different drugs in their armamentarium over 10 to 20 years. And also there's a treat to target type of approach where physicians may not want to keep their patient on that same drug or that same regimen for long periods of time. Let alone the challenges of tracking patients that are going off to school and becoming adults and going off on, operationally that they're challenging. And, from an industry perspective, we do want to get our drugs to patients as quickly as we can. I'm speaking broadly and generally, but I think we don't want to generate data that aren't scientifically robust or aren't needed. And we want to answer the questions that need to be answered. And so when we're making the case internally, those are the things that we are expected to put forward, and the types of information that internally is expected. And then finally, we've discussed this a lot, but I had this in as well, is alignment across agencies or misalignment across agencies does pose challenges. And again, these are very hypothetical examples. But if we're asked to do a randomized study with sham injections to maintain dose blinding on one side, but not on the other, it becomes really challenging to just even conceptualize putting kids through that because one agency wants it, but the other doesn't. And then a long-term safety registry that's required by one or not the other. So it gets hard to even just explaining to investigators the rationale for doing those burdensome types of studies. So alignment would be helpful in those regards. So in summary, I think extrapolation of safety is different from extrapolation of efficacy and incorporates different information in the extrapolation concept. The agency has been asking for a separate extrapolation concept for safety and efficacy, and that it does make sense. Where there's low risk of a divergent or pediatric unique safety concern, extrapolation must streamline development. We should not be doing studies in kids that aren't needed. But the sponsors need to be generating a transparent safety extrapolation concept that incorporates the data that are needed in order to make that case. And changing the course should there be a risk of a divergent safety concern. And then cross-agency alignment would be helpful, and a method for that would be beneficial. And certainly putting this in the frame of a benefit risk assessment, which for pediatric lupus, and especially the pediatric, more severe forms of pediatric lupus, that needs to be factored in as well. Thank you.
Thank you so much, Laurie. We're going to go to a different perspective now on alternative approaches and trial design considerations. We have Hermine Brunner from Cincinnati Children's to provide the academic perspective, and that would be followed by a talk from Martine Dehlinger-Kremer from UCB Biosciences to provide the industry perspective.
Before I get started, I would like to express my appreciation for the FDA team for having, set up such a wonderful workshop and listening to us, and in order to get better medication to children with lupus. Here are my disclosures. I'm gonna touch on 2 points. I'm aware it's a workshop that the FDA is our main listener, but I think we also need to use the stage as pediatric rheumatologists to point out what industry could be doing better, because in the end, we want to have better medications. So in my mind, and what I heard yesterday is that lupus in children and adults is extremely similar. We use the same classification criteria, the same response measures, the same activity and damage measures, and we even use the same treatment guidelines. But there could be differences in the maturation of organ system, and Dr. Son just showed that. There could be difference for medication that impact growth and development. And obviously, we need pediatric preparations because large paper sizes may not fit our children. So when I put all of that together, in my mind, the only gap that exists between pediatric and adult patients with lupus is for a medication where the mechanism of action might impact growth and development of a child, possibly long-term fertility. Let me make that point. As parents, we know that our children are not different the day after they turn 18. And I dare to say that between a 17-year-old child and an 18-year-old adult, there is a smaller difference than between an 18-year-old adult and a 70-year-old adult. And yet we make that artificial difference that something magic happens at 18. Now, may I remind you that up to the year 1971, you only were an adult when you were 21 years. So if you look at that, in 1970, if you were 18, you still were a child. So it really makes no scientific difference. And we heard yesterday that really organ involvement and even immunological systems are mature after the age of 5 when we would see non-monogenic lupus in children. So taking all of that together, for most situations, an open-label PK/PD study seems sufficient to show that a medication is appropriate for use in children with lupus. And a placebo or standard of care arm seems only needed if there is if we are studying a medication that could have for which there is a scientific reason that its mechanism of action influences growth, development, or fertility. Example for that could be, there are animal and preclinical data that could support that. An example could be cartilage stippling in some preclinical studies of JAK inhibitors. And if that is present, then you should look at bone growth and bone maturation. Cross-reactivity with the estrogen receptor to see whether there is a difference on progression of puberty or growth. Another issue is that really we need help both from industry and the FDA to circumvent late starts of studies in lupus. I showed you how much off-label use there is and how that hinders the enrollment in children. Really, there should be an avoidance of a deferral of pediatric studies until phase 3 programs in adults is almost completed. In my mind, the studies in children should start after the industry has made the commitment to proceed with phase 3 trials. Potentially exceptions could be, again, mechanism of action of the medication related. And in my mind, if a medication gets a breakthrough indication by the FDA for adults with lupus, wouldn't it be good to also include the pediatric population, because arguably the need in children is even larger than in adults? What we have observed and what kind of I showed you yesterday with the IV belimumab study, there seemingly is a late alignment between the FDA and the EMA in how international studies are being conducted. And obviously, we only get one shot in one study, and if it's successful, we get a pediatric indication. And if we don't get the pediatric indication, the medication may not be accessible to children with lupus. So there should be an early agreement by the for the true, not the initial PSP, by the FDA and industry, and large numbers of PIP modifications should be avoided, because otherwise there's a moving target between the two agencies. You know, what will be the plan for the study? And these protocol amendments that are then being needed really delay study start. And there needs to be an early development of a pediatric formulation so that the lack of such does not delay the onset or the start of the pediatric program. So what could industry and regulators do to help us to identify medication dosages for our smaller children earlier? Well, at least 5 to 8% of participants in our lupus studies have pediatric onset. You can do the math. And so there could be sub-analysis for medication response in adults who have childhood onset lupus, because arguably their disease hasn't changed only because they turned 18. And medication response could also be tested in adults with multi-organ involvement. We heard so much about the severity of the disease yesterday, but there are also severe patients in the adult population. So doing that sub-analysis could help us to see whether we have the same exposure effect relationship. And additional analysis could be done to look at the response of medication by body weight, body surface area, and age, also in adults, because I do think that even adults would benefit from more accurate dosages if GFR is changing, and we had some examples for that. In my mind, PK and PD and initial safety in adolescents could occur while the phase 2-3 trials in adults are occurring. How could that be done logistically? Well, there could be an extra open label arm in an adult trial where we just have children enroll, but that needs to be happening at a pediatric site. You can't just transplant a child with 12 to an adult rheumatologist and hope that they enroll in a study. That will not happen. And I also would challenge you that we need to avoid the staggered enrollment, as it leads to an interruption of the pediatric trial program and to further delay in getting medication to children with lupus. Again, an exception would be if there's a specific safety concern based on the mechanism of action of the medication. We also need to avoid organ-specific sub-studies. I dare to say that all children come with all organs. And if you have multi-organ involvement, then, there will be patients who have lupus nephritis and also skin and joint disease. So dividing them up is really difficult. And if you were to be right now in a placebo arm or standard of care arm, there will be a higher risk of flaring in these extra organ systems. And, how much more do we want to parse down the patient? Should we look at the lungs extra and the heart extra? So isolating the kidney is, really, in my mind, a little bit arbitrary. And we do know that the physiologic function of these different organs over the age of 5 in children is really very much the same as in adults. If there is a specific regulatory concern, an option could be to do some oversampling. So if you want to have more a signal what the medication could do on brain development or brain disease or on cardiac disease, you could stratify enrollment. And just to point out that 37% of children with lupus also have lupus nephritis on average. In some populations, it goes up to 80%. And that 25% of patients with lupus nephritis have CNS lupus. So how are you going to parse all of that apart? Now let's go over to eligibility criteria. Right now, industry, in order to make extrapolation really easy, they use the same as in adults, but that is not appropriate. Prior medication exposure beyond a medication that has the same mechanism of action should really not be restricted. What is the reason for it? There is no science reason for it. There's a reason for, just copying the last protocol. And the rationale for prohibited concurrent medication should really be based on potential drug-drug interaction. So what I see in many protocols, methotrexate is allowed, leflunomide is not. It's the same mechanism of action. What's the science behind it? And the answer is, well, we did it in the adult program. I don't think that that is a valid answer. Washout periods should also have a scientific rationale, and it should be based on a safety concern. You shouldn't just, if a patient if there is no safety reason to start another medication, then the patient should be started on the experimental medication right away. And any spillover effects in potential safety and efficacy should be addressed in sub-analysis rather than letting patients linger in this pre-screening and screening phase that really is detrimental to children. And there should not be a minimum duration of disease required for a child to participate in a study. They have acute onset. They are at high risk for damage. So there's no reason to let them linger for 6 months in active disease just to kind of make them comparable to adult programs. Let me put in a plug for the randomized withdrawal design. And to do that, and Martine going to talk a lot about different trial designs. But that trial design actually was advertised by the FDA in the 1970s as one of the options to study rare diseases because it limits placebo exposure. So every child who is active, it's not working for every medication. It's not similarly good. But there was seemingly some misalignment about the need for a randomized withdrawal design as compared to a parallel design between the two regulatory agencies, and that's why I'm bringing it up. So over here with a randomized withdrawal trial, every patient who is actively sick with lupus gets active drug. If it's not working for them, they drop out of the study. Only if it worked for them will they be randomized to receive standard of care or continue active drug. And then only if they have a small flare, they would basically drop out of the study and again get open label medication. The pros and cons are shown on the side. They're widely known, so I'm not going to go over them. Now, the argument why some agencies don't like the randomized withdrawal design is because flares cause damage. Now, I dare to remind you that the secondary outcome for the IV belimumab study in children was severe flare. And guess what? The placebo patients got many more severe flares than the patients who got belimumab for a year. So during that 1 year, we saw a lot of severe flare. And that's just, the science still goes back and forth. But I show you here the slide of a table from Dr. Kulmar from Monash University. And she stratified adults with lupus, whether they had high disease activity or not high disease activity. And as you can see, if you have high disease activity, that's what you have when you enter clinical trial. Your risk for a mild and moderate flare going to be higher, and the risk for a severe flare is higher. So arguably, if you use the randomized withdrawal design and get to a low disease activity, your risk for flares is lower. And mind you, high disease activity, after when they did corrections for comorbidity and organ involvement, was the prime reason for developing damage, something we want to avoid in children with lupus. If the randomized withdrawal design should be happening because some mechanism of actions of a future medication demands it, then unequal randomization needs to be carefully reviewed because it can increase sample size. There should be, or there must be, an early escape arm for children who are not doing well on the standard of care that are randomized. And these children should have the option to get active drug if it could be beneficial to them. There should be a limit of burden of study participation. So do not change the study platforms and the study vendors halfway through the study and update your portal. Do not ask for patient-reported outcomes to the excess, so the patient is fearing coming to the study visits because they do 45 minutes of quality of life questionnaires. And there must be post-study access, long-term access to the medication. We cannot do individual IND, and the patients who participate in the clinical trial and take up the burden, they deserve to have access to the medication, irrespective of their insurance status and income. Other designs are possible. I think the ones that are possible are platform trials and phase-in trials, or the randomized run-in trials where the placebo period, it's shown on the left side of the slide, is anywhere between no placebo in the beginning up to 12 weeks. But again, it really depends on the medication you're studying. Platform trials would be an option to save any standard of care arms, but they need considerable infrastructure, so I don't think that they're going to happen anytime soon in children for pediatric studies in children. So my take-home points: limit deferrals of pediatric studies and start enrollment while adult phase 3 trials are still ongoing. Omit placebo exposure in children unless scientifically justified based on the mechanism of action of the medication. The washout periods and concurrent prior and prohibited concurrent medications needs to have a scientific rationale, not one that's come up by somebody in a regulatory environment who tries to avoid any potential risk, and limit the burden of study participation for families and sites. Well, thank you for your attention.
So good morning, and I would like to thank the FDA for organizing this excellent workshop and also for the opportunity to participate. I will give you an industry perspective on ways of doing pediatric studies in SLE. So my disclosure, what I would like to share with you, some background, study designs options that we have, the integration of pediatrics in adult trials, and conclusion. As we all know, SLE is complex disease and we need long-term outcomes. What we have is a lot of data in adults. We have extensive information, but in children, the prevalence is lower and we have a limited number of patients. And we heard yesterday, we have a high unmet medical need. So we have a large pipeline of SLE in adults. We need really some pediatric therapies. Important is to involve the patient and patient organization in the development of those therapies. Just to show you how many pediatric development plans are out, that is from the publication from 2025 from Weber to Delisa. There were 22 PIPs approved yesterday. Weber 2 provided updated data that at present time we have 26 pediatric programs that need to be fulfilled, and 10 of those programs have stopped because the investigation in adults has stopped. And this shows also the complexity of SLE disease because numerous products are not continued. And so we are in a situation where we need optimal and study design in order to conduct those studies. There is no one-size-fits-all solution. Options can range from randomized controlled trials, such as the landmark endpoints or withdrawal that Hermine just presented, to more flexible approaches, such as platform studies, the inclusion of adolescent and adult trials, but also single-arm designs, as well as full extrapolation supported by BK matching. The Bayesian extrapolation approach enables justified borrowing of adult data to support pediatric efficacy claims, and this reduces the number of pediatric patients that we need. The Bayesian extrapolation approach is part of some of the design, an intrinsic part where we have the solid lines, and it is optional for the designs that show here with the dotted lines. I will focus now on the most innovative designs, and we'll start with the design where we need the least number of patients. So full extrapolation of efficacy from adult supported by an open label PK matching or PK-PD. And this design represents potential full efficacy extrapolation approach, which is supported by PK comparability, with PD comparability providing additional support where relevant and available. In that case, we enroll pediatric SLE patients with an active disease into an open label treatment, 12 or 24 weeks. We do an PK interim evaluation for dose confirmation, and this will give us also some information about safety and tolerability. The primary endpoint will be PK, the secondary endpoint PK, PD when relevant, safety, immunogenicity, and clinical evaluation. And for the safety, we would include the patients in long-term open label extension study. Opportunities and advantages of this: it's highly accepted by patients and caregivers, very small number of pediatric patients that we need. From an operational aspect, it's really feasible, and it accelerates the development of the drug, so gives access to the patients very fast to have the product available. But what we need to consider is that it requires strong justification that the disease biology, the treatment response are sufficiently similar between the adults and the pediatric patients. PK comparability must be demonstrated. There are some residual uncertainty which remains regarding the magnitude of pediatric efficacy, and we also need to consider the potential difference in these phenotypes and baseline characteristics. As we discussed already yesterday, we have this difference of activity and also the lupus nephritis, which is more prevalent in pediatrics. What I just presented to you was the simplest extrapolation approach, demonstrating that children achieve similar drug exposure to adults through PK matching. That approach relies heavily on the assumption that similar exposure will translate into similar clinical benefit. In some situations, however, PK matching alone is not sufficient and does not provide sufficient reassurance. And in that case, we may want to have additional evidence on PK activity, clinical outcome, or some form of efficacy comparison. And this is where the single-arm hybrid designs become relevant. But we start from the common foundation, which is PK-PD, regardless of the design. We first want to understand whether the drug reaches the target and produce the expected biological effect. Then next comes the question about how efficacy is translated. And at this point, the design begins to diverge. Some attempt to infer with no control and other with control. We have, for the control, we can have a clinical threshold control, we can have synthetic arm as control, or we can have digital twins as control. The four approaches have different ways to make that efficacy inferred. And if we go to the exposure-response relationship, we have a higher exposure, which shows better outcome and thus drug efficacy. For the clinical threshold, we compare against a threshold, and treated patients must do better than a fixed clinical relevant threshold. If we use a synthetic arm, we compare against observed external patients, and the treated patients do better than similar real-world patients. And for the digital arm, we compare to predicted untreated outcomes, so the patient improves compared to what would have happened without a treatment. What data are needed for these four approaches? We always need the standard, which is the treated individual patient data, plus biomarkers and outcome. For the clinical threshold, we need in addition an agreed threshold. For the synthetic arm, we need external patient data, such as registry, for example, real-world data. For the digital arm, we need rich longitudinal and modeling. Each of those approaches has its strengths and limitations. For the exposure relationship, we have fully mechanistic. It works very well when we have small population. It's strong for dose justification and extrapolation. On the other hand, we have no direct comparator, and we rely very heavily on the biomarkers and extrapolation. For the clinical threshold, it's most intuitive, no need for external data in that case, but we have to rely on the expertise and the clinical threshold that we have set. For the synthetic arm, it encodes to real patient data. It's more intuitive and it reduces the reliance on the biomarker. But the comparability issue can come up because we really need ideally IDP and strong adjustment. For the digital twin, it's fully individualized and it handles heterogeneity well, what we have actually in SAE, and we don't need to match patients. On the other hand, this approach relies heavily on modeling assumptions, and it might also be difficult to validate. When can we use each of those designs? So no control. When we have strong adult data, we have a strong biomarker, which is well validated, and very small patient population. The clinical threshold, we have no reliable external data that we can count on. We have a sample size that is very small, and there we have a clinical threshold that we all agree on, sponsors as well as authorities. And for the synthetic arm, we need good registry data and strong comparability evidence. For the digital twin, it's good when we have an heterogeneous disease, but we need really rich natural history data, and we want individualized inference. Why are those designs relevant for pediatric SLE? We have strong biomarkers and mechanismic understanding allows exposure response inference. For the clinical threshold, sample size is small. We have clinical meaningful benefit, which may be more informative than a direct comparison. And we can use a synthetic arm. We have a rare disease. We have external data that can replace or reduce the need for controls. And for the digital arm, we have a high disease heterogeneity, and this approach favors individual counterfacts. The overarching message is that the characteristic for SLE, including its rarity, the limited patient availability, the disease heterogeneity, and the potential to leverage relevant adult evidence through extrapolation, create a strong rationale for considering innovative single-arm hybrid approaches. The Bayesian extrapolation design. The Bayesian dynamic extrapolation design is an important concept for complement many of the study designs relevant for SLE. It's a statistical framework that allows us to incorporate information from another source, in general, the adult, while still allowing pediatric data to be determined and to determine how much information should be actually borrowed. When adults and pediatric, so instead of saying pediatric effect equal the adult effect, we say that the pediatric effect is around the adult effect with some uncertainty. And it is the model which learns how close they are in reality. And the closer they are, if we have an agreement between the adults and the pediatric, we borrow a lot from the adult data, and the less close they are, the less we borrow. And by borrowing strange from adult data, The sample size is reduced, and so this design achieves a streamlined study, and we can support regulatory as well as efficacy. We can support efficacy, which with a clinical endpoint. So the Bayesian dynamic extrapolation aligns particularly well with SLE because we have extensive adult information. We have a substantial biological similarities between adult and pediatric disease. The clinical endpoints are broadly aligned, and we have a limited number of patients, making large trials difficult. The goal of this design is really to reduce the amount of pediatric efficacy data required, while at the same time maintaining confidence in treatment benefit. Another option that we have for a pediatric SLE, and it was approached by Hermine, is the platform study. For the platform study, we assign the patients with an active SLE to drug A, B, and C, and we have only one share control. This is very relevant because, as we know, as we have seen, we have a lot of products in development in SLE. This is a design that is also very much accepted by patients, and they don't have to choose. We have one protocol, we save in terms of controls, but the point here is that it needs to be set up. And how can it be set up when we have different competitors? We have seen successful platform trials in oncology, and they were set up as certain organizations. So an interesting way of doing studies, of accelerating the development, but the question is, how can we set it up? The further option to accelerate and give faster access is to include adolescent and adult trials. For this, certain prerequisites have to be fulfilled. The disease must exist as a continuum between adults and adolescents, so no significant difference in PK responses. In terms of PK, we need characteristics in the adult patients and expect it to be similar in the adolescent. And the efficacy and safety data from the adult patients must be such that we don't put the adolescent at risk. This approach is very much supported by ICH E11. And if we go this approach, it's important to discuss it with the authority upfront. The advantages: it's an accelerated development. It reduces or eliminates the need for further standalone studies in adolescent, faster approval for adolescent, and it gives access to innovative therapies fast. And it also reduces the off-label use. From an operational aspect, there are also some efficiencies. We have one protocol, and we can share infrastructure. From a statistical power and robustness, larger combined sample size, which improves the precision of efficacy and safety estimates. And we have also consistency of data, regulatory alignment. As I mentioned, we have ICH E11, which is very much supporting and say that we should include the adolescent in adult trials or do parallel adolescent-adult trials whenever that is possible. There are, of course, some points to be considered when using that approach. So the endpoint suitability, we have the same endpoint for adolescent and adults for SLE, but we can have, for example, the primary and secondary the same. We can say that the tertiary endpoints could be optional for adolescents to make the trial more attractive. We need to capture adolescent-specific outcomes such as growth and development. SLE quality of life tools are validated in adults. We don't have validated ones in children, but we can use, for example, among others, the fatigue quality of life. The safety is important. Adolescents might have different AE profiles, so to perform a separate safety analysis. The psychological and compliance challenges is to be taken into account for the adolescent. We need also to look into logistics. So an adult site will not recruit an adolescent. So also look into this to have specific sites, and also to look at the transition from adolescent to adults in case the transition happens during this study. And those studies would then be part of pediatric plans, so important to discuss this with the authorities in advance, and that might affect the phase 3 design. Having regulatory approval is not sufficient to get the drug accessible to the patient. So from the beginning, we need whatever design that we choose, we have to take into account that it must be relevant for the health technology authorities and the payer. So we need to fulfill also the requirement of those authorities. So look into comparative evidence, patient-relevant outcomes, methodology rigors, real-world relevance, and discuss early on with HTAs in order to get the alignment so that the product can also become to the better of the patient. I may conclude the similarity between adult and pediatric SLE enables early adult data to guide pediatric clinical efficiency. The rarity of pediatric SLE and the competitive landscape that we have limits the traditional study designs and requires innovative study designs. Early engagement with patients and families or caregivers is important so that we have optimal study designs, and alignment between regulatory authorities is also important so that we have pediatric development in SLE and we can accelerate the access to the therapy. Important is scientific innovation, patient engagement, regulatory collaboration, and if we put this all together, we can accelerate and get pediatric SLE therapies developed. So I would like to thank also my colleagues who have supported this presentation, and thank you very much for your attention.
Thank you so much, Martine, and thank you to all of our speakers this morning for helping us stay not only on time, but early. So this is going to allow us to take a slightly longer break. So we'll go ahead and break now, and if we can ask you to return at 10:30, the kiosk will open, but at 10:15. So take your time. Online participation, we have many online participants today as well, and we encourage our online participants to use the chat to pose their questions, and we will address them during the panel discussion. And we are now moving with the panel discussion, and I would like to invite the moderator, Raj Nair, to come forward, as well as all the panelists.
So I want to thank everyone for continuing to stay in the workshop, and I know we're coming towards the end, and I appreciate everyone's participation. I think we still have a couple of panelists that haven't had an opportunity to introduce themselves, so I'd ask that you introduce yourself, and then we'll take the questions after that.
I can start. I'm Sarah Ringold. I'm a pediatric rheumatologist, currently at Johnson & Johnson, a director on the pediatric development team. I work across indications in pediatric rheumatic diseases, including lupus. Thank you.
Hi, Lynne Yao. I kind of introduced myself yesterday, but I'm the director of the Division of Pediatrics and Maternal Health at FDA, and a pediatric nephrologist by training.
Okay, thank you. So we're going to start with the first question being the patient.
Right. I'm online. This is Kalyani Apana.
Oh.
Maybe I'll introduce myself.
So Caroline online?
Kalyani. Yes. So, hi, it's a pleasure to join the panel.
Kalyani. Do you want to introduce yourself?
Yes, please, briefly. So I'm Kalyani Apana, head of biostatistics at EMD Serono. Okay, and my work sits at the intersection of.
I'm sorry, we can't hear you in the room.
I can barely hear. I don't know. There might be an audio issue.
You can't hear me?
It's very faint.
Okay, so. Why don't you continue? I'll try later. Let me see if somebody can fix this. Maybe Caroline can try and see whether it's working.
Okay.
Yes.
Okay. I apologize. So, okay, let's get started with the questions. So we start with the patient family question. When considering enrollment in a clinical trial, what information would you need to feel confident in that decision?
I would need to know what to expect as far as the side effects and how they may impact my everyday life. So when it comes to academics, extracurriculars, and maintaining my social life, I would also need clear information on the likelihood of the trial drug triggering a flare, and I would also need full transparency throughout the entire process.
And then what kind of risks do you feel you would be willing to take if it's already approved in a in adults?
For a drug that's already been proven to be safe in adults, I'm willing to risk having reasonable minor side effects and taking time to adjust my dosage if needed. But that being said, I would need those risks to be weighed against keeping my lupus managed so that it doesn't negatively impact my academics.
Okay, and so if it was approved in an adult patient population, how confident would you feel that it would work in children as well?
I feel like I would feel the most confident for treatments that target the symptoms experienced similarly by both children and adults. And I would need strong data on how adults respond to the treatment, and that justifies extrapolation.
Okay, thank you. Okay, so does anybody else want to add like any thoughts on those questions? No, we can move forward. So for the extrapolation of adult efficacy to pediatric population, there exists a continuum, and with the degree of additional pediatric data required depending on the nature and magnitude of remaining gaps and uncertainties. So is there any additional data that should be collected in adults to support an efficacy extrapolation strategy? And I'll open this up to the whole panel to discuss. Lynn or Stephen?
Sure, I'm happy to get us started. Yes, so thank you again for the discussion. And so I was sort of reflecting yesterday on, all these fantastic presentations we've had. And, I think the data so far really suggests, similar disease, but I know a lot of questions came up yesterday in terms of, is there really similarity in response to treatment? So I think relevant to this question is, do we feel like there's a similar enough response to treatment that we can leverage something like exposure matching, or is there additional data needed from an exposure response or PK/PD perspective? So I wanted to offer maybe a little bit of commentary after reflecting on yesterday's discussion, and then how we can maybe leverage some adult data to help fill that gap. And so when we think about how we practice pediatric rheumatology treatment right now, there's almost no drug I can think of where we are dosing children higher than what we see in adults, right? So if we think about hydroxychloroquine, 5 mg per kg, typically max of 400, the same thing we use in adults. We think about cyclophosphamide, same weight-based dosing, methotrexate, maximum of 25 milligrams, again, not exceeding adult dosage. Mycophenolate mofetil, 1,500 twice a day, again, not exceeding adult dosage. So the community right now is not practicing under the paradigm that children require a higher exposure to achieve response. And so if that paradigm is not true, so one, it totally reshapes the practice of pediatric rheumatology at the moment. But two, again, it's not necessarily in line with the data, albeit limited data that we discussed yesterday in the exposure response section. So I think the point, though, is very well taken that patients with higher disease activity may require potentially higher doses. And so to me, this is really a point for the adult program. So it's really important in my mind for there to be really robust exposure response data in the adult programs. And then I think that would support exposure matching. But these adult programs really do need to look at patients and see whether disease activity or organ involvement are covariates, right, on the required dosing. So I think this is really sort of a fantastic way to leverage adult data, but we need to be forward-thinking in that.
I had a follow-up question to that. So I guess while there isn't a ceiling dose that's being exceeded, is there any increased frequency of dosing for any of these products in kids? Like, do you see maybe remanifestations faster such that you would treat more frequently? Is that happening?
Yeah, that's a great question. When I sort of think of dose and dose frequency, sort of thinking about those in terms of exposure, yeah, I still think in terms of whether we look at dosing interval or absolute dose, it's still pretty similar between adults and children, at least how we're practicing now. But, along those lines, I think one of the important things, the academic communities can sort of contribute is, remember, we approve drugs sort of on a population level, but we do as clinicians know that individual patients' responses do differ. And I love that regulatory authorities have already been forward thinking about this and a number of other diseases, right? So we know rheumatoid arthritis with adalimumab. Some patients require a higher dose, which is reflected in the drug label. Children with FMF or TRAPS, again, potentially a higher dose of canakinumab is recognized in the label. Secukinumab for psoriatic arthritis. And so we know that even once a drug gets approved, as clinicians, as academic rheumatologists, we have to understand how to optimize these doses in clinical practice. And so that's the burden on the research community. But again, I think the major role for drug regulators is to recognize that for labeling purposes, right? Because if that's not recognized in the label, then we can't access it for insurance reasons. Our patients can't access it. So hopefully that perspective is helpful.
Thank you. So I'm going to try and see if we've got Kalyana, whether we can hear her. Kalyana, do you want to try and speak? You're on mute right now.
Yeah. Can you hear me now?
We can hear you now.
Okay, so maybe should I add something to what Stephen just mentioned?
If you can introduce first and then, yes.
Oh, okay. So my name is Kalyani Apana. First of all, pleasure to be here, part of the panel. I'm the head of biostatistics at EMD Serono, and my work actually sits in the intersection of statistical science and drug development, leading the design and interpretation of studies that establish whether medicine is safe and effective. So nice to meet everyone.
And did you have any additional comments you wanted to add?
Yeah, so just following up on what Stephen just mentioned, I think what is also important is to maybe conduct pre-specified subgroup analysis in the adult SLE trials. This will enable us to assess whether there's a treatment response. So this kind of subgroup analysis we want to perform is to really assess whether there's a treatment response in adults with childhood onset SLE, the response in adults with multi-organ involvement, And also, the response by weight, maybe body surface area, and also age groups. So these analyses will then directly inform whether, adult data can be extrapolated to the PEDS population and should be initiated early and not really wait until after the phase 3 completion. So this is just, I have some other points that I can bring up later, but this is just a follow-up to what Stephen just said.
Thank you. And so I guess the next question here is, oh, yes, of course.
I am very swayed by the conversations and the discussions that we've had the last day and a quarter or so. But one of the things that I do wonder, I recall back in the PJIA workshop that we had, that we had rheumatoid arthritis that was called a different disease from what was in children, which was PJIA. And we were saying, well, these are two different diseases. And what we learned from that workshop is that the real reason that those kind of became different nomenclature, even though it was the same disease, is that there were different people taking care of them. They had the adult rheumatologists who were taking care of rheumatoid arthritis and pediatric rheumatologists who were taking care of patients with idiopathic arthritis. And so they kind of came up with different names, even though the diseases were very similar. I do wonder a little bit, are we kind of in the same situation here? As a pediatric nephrologist, I took care of a lot of pediatric lupus patients, but I only took care of their nephritis. And, they would be on methotrexate or Plaquenil, and I'd say, okay, fine, whatever. But, you have nephritis, so I'm going to treat you with what you need to be treated with for the nephritis. And so are we talking about a condition that is, again, multi-system that can infect many different organs, but that we seem to have siloed these treatments because it's different people who take care of it? I don't know if that's part of it, but I do think that to answer that question directly, what additional data should be collected? I think we need to rethink who we're enrolling in the adult trials that study lupus, and do we really need to exclude those patients with nephritis? Because if we include those patients with nephritis, I think the remaining uncertainties I have about whether or not the response will be different will be completely addressed.
Well, then I think your question is very apt about the concept of different providers treating different patients. As someone who crosses those lines, I think I have, and Stephen and others who have spoken, have like kind of firsthand experience of that. As Stephen pointed out, it's really the same medication usage is happening. In my opinion, I think Stephen alluded to this in his initial talk, there is some difference in behavior around the use of steroids, I would say. The pediatric community is a lot more aggressive about the frequency, quantity of steroids that are used. I think in part that is because children have fewer comorbidities and it's more feasible sometimes to deliver that. You have a patient who's diabetic, hypertensive, that becomes much more challenging to do. So there are differences like that, but I think the fundamental approach to treatment is really the same.
But Stacy, would you also say that in addition to more aggressive use of steroids, that overall pediatric rheumatologists tend to treat more aggressively and tolerate less active disease in their patients? I think that's certainly seen in the JIA conversation you mentioned, but I think across the board, pediatric rheumatologists tend to tolerate less.
Yeah, I think to a certain extent that is true. I think some of that depends upon the adult rheumatologist treating the patient as well, but we are more aggressive. Again, I think that again comes into play are the comorbidities, right? I mean, you have an adult with multiple comorbidities, and the aggressiveness of your therapy is going to have impact that you're not going to see in a child.
Yeah, I would agree with that. But again, we should not misconstrue the aggressive treatments are because children necessarily require it, right? It's just potential different goals and comorbidities playing into that.
I would like to add on what was said before in terms of sub-analysis that should be done in adult data. Beyond what was already mentioned is data should be collected to identify those study participants who had their disease onset during childhood. Conceivably, when they cross this age barrier, whatever it might be, the disease should still be the same and shouldn't fundamentally change by a birthday. So that would be something else that should be considered. And with your comment about the different nomenclature, I think in lupus, we are in a much better situation because we use the same classification criteria, the same nomenclature to assess the activity of the disease, the damage, and we use the same treatment guideline. So the similarities have been recognized by the academic community already.
Well, I was just going to say, Hermine, when you presented your slides this morning and we had the conversation yesterday, it did make me think, I don't know, Raj, maybe I'm going to ask you, but how was it that we ended up excluding nephritis from, you know, the clinical trials that were evaluating these lupus therapies?
I would say that's pre my time, but I'll try to answer. So my thought is that the initial treatment of adult lupus nephritis, like when we're talking about severe lupus nephritis, is it's different, but it was much more standardized back then. And so my guess, and again, I don't know, but my guess is that because of what would considered be the initial treatment for systemic lupus, more generalized, versus an active lupus nephritis patient might be different, that you would definitely be including cyclophosphamide at the very beginning, or CellCept or something along those lines at the very beginning, whereas patients enrolled into more general SLE adult trials may have started on something like hydroxychloroquine or azathioprine. So my guess is that might have been why this distinction was made, but that's just, you know, my thinking on how this happened. Also, the other thing, I think I mentioned this yesterday, is enrollment criteria were probably different. Also, the response criteria so like a response criteria for general SLE is very different than what's used for lupus nephritis. Lupus nephritis, it's a complete renal response, which wouldn't apply to general SLE. So all of these kind of things, I'm guessing, are what made that kind of distinction early on.
Just to follow up on those points about background therapy, completely acknowledging that's true that there are different treatment guidelines for lupus nephritis and non-renal lupus. However, one of the things that we've seen from the presenters is that there are some differences in background therapy with childhood onset lupus because those patients tend to have more severe disease. Cyclophosphamide is not exclusively for nephritis. We also see it used in CNS lupus, which may be more common in children. There's a lot of use of B-cell depleting therapies for hematologic manifestations, possibly because of the experience with it for ITP in the pediatric community. So we do have to contend with those background medications in some of the childhood onset lupus population when doing a non-renal lupus study as well, which I think also kind of supports the argument that in some circumstances where there aren't huge expected PK differences between a nephritis and non-nephritis population, it may be more appropriate to study those together.
Yeah, and I didn't mean to say that, you know, I would agree that you got to have a line there. Like you're not going to include, you know, 4, 5, and 6, you know, class 4, 5, or 6 nephritis in you know, initial trial for an overall lupus. But if there's if there's evidence of nephritis that, you know, you have that is, you wouldn't necessarily even, you know, need Cytoxan for, that those, that not precluding enrollment in those cases.
Right. And I think that's what we do right now, actually, for general SLE, is we do allow kind of that lower disease burden in general SLE trials. I think Roberto and Caroline have their hands raised. Roberto, I'm not going to force you to keep your hand raised for 10 minutes this time.
Thank you so much. One point, there are very many interesting points in Hermine presentation that every point deserves a further discussion. For example, randomized withdrawal trial, and considering that, of course, everyone is agreeing that we should leverage the efficacy data in adults, this would confirm, in my view, of course, maintenance of effect. How to reconcile, though, a different design conducted in two different populations is something that needs to be a little bit more discussed, like how do the data complement each other, if this aspect was not considered before. But another important point, and I think Dr. Conklin, yes, was with us yesterday, announced that the anifrolumab trial will conclude early. It's an appeal like to please share the data, make analysis, do analyze the adult data in comparison with those found in children, because it would be extremely useful for the community. Whereas we have many products in the pipeline, I'm not up to speed in knowing what is concluding first, and every new example beyond the belimumab one will enrich our knowledge and understanding and help shape the future requirements for future trials. Thank you.
So I'm getting a little head shakes from the audience, so I might ask for some audience participation as far as the anifrolumab experience. So do you want to?
Yeah, I think that, oh, do you want to comment, Sheila? I think that may have been stating the wrong drug
I don't
name.
know actually which study you were referring to, but I don't expect any studies concluding earlier than expected for anifrolumab. So, you know.
I think it was anifrolumab. Was it mentioned yesterday that you would be concluding soon? Sorry for that.
No, we didn't make any comments on early or otherwise ending of anifrolumab studies. I believe there was a presentation that hopefully the slides will still be available for folks that showed what's publicly available on clinicaltrials.gov. And there was a discussion of the conclusion of some other programs, but not anifrolumab or AstraZeneca studies.
Caroline, I see your hand is raised.
Yes, thank you. No, I was trying to jump on the issue of why we are excluding lupus nephritis from trials, whether this is appropriate or desirable, especially for pediatric patients for whom we know that the prevalence is much higher, but also probably in adults with very active, severe refractory lupus, for whom also the prevalence is high. And I was thinking that this would also depend on the mechanism of action of the test drug. When it is supposed to be disease modifying, when it is an anti-CD20, when it is CAR T cells, I think we should not exclude them. And of course, the outcome measures will be different and specific for lupus nephritis. What we have seen for two CAR T cell products that we have that applied for a PRIME designation, which is some request to be included in some future accelerated assessment program with very thorough accompaniment in the form of scientific advice at each key milestone. In those two dossiers, they presented preliminary efficacy data. It was for severe refractory active lupus with or without lupus nephritis. And the preliminary data they showed were collected in trials where they included both, probably with a stratification, even if there were two early phase products to be powered strictly for this efficacy. But we could have both assessment of the general cutaneous lupus features with the standard DORIS criteria, remission criteria, or low disease activity criteria, etc. And there was also assessment of complete renal response for those patients with lupus nephritis. I think this might be possible. And when it comes to considering that we should collect data in adults as early as possible, with a view of using them to build a model for example, exposure response assessment in pediatric population and at least partial extrapolation. then it would be interesting, I think, to include them. That was my my contribution for this question.
Thank you. So oh, go ahead.
Well, one barrier I see would be that different dosages need to be used for lupus as compared to lupus nephritis because the pharmacokinetics are different based on the medication. So if one starts the study in children, it'll be really useful to know what is a good dose in adult lupus nephritis, just to make sure that these dosages are considered when children with active lupus nephritis are included.
Thank you.
Yeah, I would agree with that. I think the other pediatric specific, maybe not specific, but more common in pediatric consideration is that children who may not have active lupus nephritis at the time of trial enrollment may still be at risk due to the natural history of the disease, that they may continue to accrue those manifestations. When we see a child in clinic in the first 3, 6, 12 months of their diagnosis, we don't necessarily know what path they're going to take, what shape the lupus will take in terms of what end organ manifestations will be involved. So there is a certain risk to classifying someone as a non-renal lupus patient, not yet knowing if that's going to be part of their disease course.
Yeah, I'll add that's true for adults as well. You know, so we have to remember that adults, even with well-established disease, can still develop new onset lupus nephritis as well.
So I kind of feel like we're on this topic, so I kind of want to see if there's any additional considerations with this question. So it's, you know, about how we should use inclusion criteria in adults to define and enroll a population reflective for a extrapolation framework. Go ahead.
So what I heard from the discussions are that it would be really useful in increasing the confidence in an extrapolation approach for children if we had, number one, we had some patients and adults who had a degree of nephritis that we could assess the treatment effect. Number two, that we had a sufficient number of subpopulations enrolled. So whatever that looks like, childhood onset into adulthood, I think even just a reasonable number of younger patients enrolled. So somewhere between 20 and 35, for example, that group of patients and their treatment response, I think is very hard to say would be different than a 16-year-old, right? Patients who have multi-system disease, which is what we think more children will have. So all of these things that if we're enriched in the adult trials, or at least not excluded in the adult trials, I think would really help to, on a drug-by-drug basis, help us to say, for this drug, you've collected a lot of information. We have very high confidence that these populations are similar.
And harping on the eligibility criteria, I think there are some that are really scientifically justified, and there are some which I consider rather arbitrary, because if you ask anybody from the drug development team, they look to the regulatory team to get an answer for it, showing me that the scientific rationale is modest at best. So in general, these eligibility criteria need to be reviewed, because I also think that in order to make reference or to get the information from the clinical trial and make it useful for clinical care, we need to have populations that are representative. And this highly restrictive and regulatory subpopulation that are enrolled in a clinical trials are not that. And I dare to say the confidence of the patient and families in the result of the clinical trial would be much higher if the patients that are enrolled in the clinical trial are similar to them and not specific to, you know, a certain lowest risk study.
Go ahead.
So this, to your point, Hermine, and this is where I think Nia is extremely important. So what we've heard in other workshop settings is that having patients involved in early in the development of the clinical development program, including the protocols, doesn't just increase, the idea that you're really doing measuring and doing study measurements that matter, but also helps with recruitment. Because what I've heard is, you go back to your center, wherever you're getting treatment, you say, this patient is saying, well, I don't know if I should join this protocol. And you say, Nia, look, I was involved in the protocol. I know this is an okay study to join. It increases and helps with enrollment of populations that maybe have been underrepresented.
Yeah, I think that's a great point. And I think that's something that I'm hoping we're going to be seeing more of is patient participation in protocol development. Go ahead, Elvina.
Just another question about the eligibility criteria. Panel's thoughts on inclusion of adolescents in adults SLE trial?
So most of the pediatric rheumatologists see patients beyond the age of 18 years. We are very hesitant to transfer care of an 18-year-old to an adult provider because we have a more regulated and secure setting and a better way with social worker approach. So if you do an adolescent study, do not limit the upper age of inclusion at 18 years. Allow us to enroll from 10 up to 25 or 26. Especially patients, very often patients who are recruited are those that are treatment refractory. And these will be the ones who stay in pediatric centers longer.
Just operationally, I think it's critical for those types of trials to not conduct them in a completely adult environment. I think if you're sending an adolescent to participate in a trial that includes adolescents, they're going to be very unwilling to change sort of their care setting to participate in a trial. So it is an investment to include pediatric centers and sites, but that would be required.
Yeah, sorry, I was just going to comment that I think it was covered nicely in one of the earlier talks about different strategies for perhaps doing this, including a separate open label cohort. If it's a randomized placebo-controlled trial, may not want to put adolescents into that same design. So considering this separate cohort, and I'm also interested if the adolescents are enrolled into the adult trial, what it then looks like for the younger patients, if that becomes a smaller, given that those patients are more rare, harder to enroll, harder to find, then you interested maybe from a regulatory perspective, what the study then looks like for the younger patients, if that's then a second cohort that's much smaller, that just PK/PD, or what would be accepted from that standpoint.
I'm sorry, Sarah. I'm having a hard time hearing you. I don't know if it's something to do with the speak.
Did other people hear me?
Do you want to repeat it? I think it has something to do with where I'm standing.
Okay. So I think I was mostly reflecting on how the how the topic had been covered before and some of the strategies that were discussed for different ways that the adolescents could be enrolled and not just put into a placebo-controlled trial that was designed for adults. And then I am also asking about maybe from a regulatory perspective, then what it looks like for the younger patients, the 5 to 12-year-olds, and what level of data is then accepted for these younger patients who are more rare, perhaps more difficult to recruit. What level of data or what the study then looks like for them.
So, I mean, I think that's a great question. And I think, that's part of what we're hoping to get answered here is, what kind of data do we need in the full 5 to 18 patient population, whether we need the same level of information in the 5 to 12 if we have 12 to 12 information in 12 to 18-year-olds, whether that's whether we need similar information or different information. Hermine, I think you have a question.
Again, we are sliding back on the concept that the organ function of an adolescent is different than that from adults. There is no indication. We saw all the data yesterday from immune maturation of the immune system, cardiac function, renal function. It's comparable. So if there was an adolescent, I dare to say it shouldn't even be an adolescent arm. It should be an arm for also the younger children, because that is where the data are missing, where the extrapolation may be more limited and where the safety profile could be different because of the different risk for infection. So if there's an added-on arm, maybe it should cover the entire pediatric age range, and one could use, the adolescents that are enrolled and could, analyze them together with the adults in the same trial that had childhood onset disease. So you would have a relative larger body of evidence.
Lily, I see you're up.
Actually, my question was related to the point that Hermine just made just now and earlier as well, with a call not to defer studies in pediatric patients. And the point that was made by Vikram Sana that we can reasonably predict dosing down to 5. So maybe a question to our industry colleagues, is it feasible to have one formulation that would accommodate patient down to 6 at the time of the adult phase 3 trial? And also the need for additional juvenile toxicology studies to support enrollment of patients 5 to 12 years of age so that we can accommodate this approach of opening enrollment down to 5 instead of deferring studies until approval in adults.
Yeah, I can start. That's a good and big question. I mean, I think we've heard from a lot of our patient representatives that they would be more willing to take a degree of risk when there's still unknown information in terms of placebo exposure, which I think makes a lot of sense. And we also heard from Emily's talk yesterday about really nicely describing this period of time when we want to study in pediatrics between we have enough data for a positive risk benefit, but not so much that everyone's already using it off label. And I think a lot of what we worry about timing wise is does that window still exist and long enough to really complete a trial well with the data that Hermine had showed from off-label use really starting quite early in some cases before the pediatric programs. So I think in terms of whether formulation can be adequately developed prior to a phase 3 program down to age 6 is probably going to be very drug dependent, both in terms of the mechanism of action and the anticipated on and off target effects, as well as the PK and whether that is expected to differ because of the formulation of the drug. So I don't know that there's a universal answer to that, but I think the point is well taken that if there is suggestion of benefit in phase 2 or earlier trials, in many ways, it makes more sense to get that robust data from some younger participants earlier in the trial versus when we already have large adult trials. And from, I think, everything we've heard from the panelists, really strong indications that this is not a different disease and that children are likely to experience the same benefits. Because I think we can all agree that the current paradigm of starting a pediatric trial that takes 5 years or more after phase 3 raises a lot of both feasibility and ethical issues for those involved.
Yeah, and I'll just add, I think for enrolling adolescents into adult trial may be the right solution for a particular study or drug product, but in general, we have to ask ourselves, how much data is enough, right? So if we think back to yesterday's session in the ALMS trial, where 25 adolescents were enrolled with lupus nephritis into the adult trial. We looked at that data. I'm glad we have it because it was helpful. But was 25 patients enough for us to feel confident that children respond similarly to adults? And I think one of the challenges with enrolling adolescents into adult trials is the sample size is going to be fairly small, right, with adolescents. And so do you gain enough data from that to make it worthwhile, realizing that the adult trials, again, are more likely to have placebo exposure? And so why potentially expose those adolescents to a more complex design, potentially higher risk, if we're not going to have the power to really look at that subgroup? And so do we really gain more from enrolling an adolescent into a clinical trial versus adult trial versus doing like a larger open label PK/PD safety study?
Go ahead, Lynn.
So we have these kinds of conversations like almost every day at FDA. And that issue about when is it right to enroll adolescents into, during phase 3. And to your point, exactly, Stephen, we have this sort of tension between, well, we can enroll into an adult trial, or we could wait, and then if the efficacy looks good, all we really need are PK. And then you say, okay, well, then we're stuck. Do we wait so that we can get PK, or do we go ahead and enroll them? But I think what we tried to discuss in the E11A guideline is that the concept of enrolling adolescents during phase 3 doesn't always mean that you have to enroll them in the adult trial. That what we could do is collect data we think are necessary to support approval during phase 3, such that we don't have to wait. Now, sometimes that will be enrolling adolescents into the adult trial, but it could, and we could imagine that might be something different as well. So I don't want the group here to think that if the opportunity to get adolescents or even younger patients, we'll just throw it out there, right? The whole population in with the original approval depends on, what we think is what data we're going to need. And we have conversations every day about, well, if 25 is what you need, how is that different from 10? And then how is that different from 5? And then how is that different from 0? So we have that conversation every day. And it's really important that we think about, what information are we gaining? What are we getting from that? And if it's safety, and if it's, some question that we're answering, I think what we just want to hear is justifying what that number would be.
So I wonder which nomenclature we use to define an adolescent. Do we use the WHO ones, and that's why, so they turn 12 and then they are adolescents? Or do we use some other nomenclature that's used by the pediatric community, where we say adolescents are, they are early adolescents and mid-adolescents and late adolescents, and then we will go under the age of 12. So when we talk about adolescent enrollment, it should be a rationale why we limit it to adolescents. It could be a safety issue, but if there is no safety issue, what is the issue, or what is the reason why we deprive children who have early onset disease, and where we know that the higher genetic load and the higher life expectancy and are much longer at risk to be exposed to chronic steroids? Why would we deprive them from medication that could change their lives and enable them to get the full potential and growth and development? I can't see it.
Yeah, so I completely agree with you, Hermine, about this age as a sort of arbitrary biomarker. In fact, age is, we've said this at FDA many times, but age is actually a very, very poor biomarker. Like, it's a regulatory and legal definition, but it's not really a good biomarker. We just use it, from a regulatory perspective. What we would like to see, and I think what you're saying is, just give us the justification why scientifically or clinically this is the population you need to enroll. Is it 9 to 25? Is it 5 to 25? What is that population? And what do we have to support that it's okay to put them in phase 3?
Just to add on, when you are in an Arabic country and you turn 12 as a female, you will be considered an adult patient. We would not have that same, so it's a legal definition, and everything in terms of inclusion, such a patient who has not legally, is not legally able to consent. That's an research ethical question, but not a regulatory question in my mind. The consenting needs to be done properly. The assents need to be done properly. If you go to a different country, go to Canada, if you participate in a clinical trial as a 16-year-old or even as a 14-year-old, you have a say. because it's your body. So we need to kind of look at it in a different round between the science we need to show for drug efficacy and the ethical part of participating in a clinical study.
So all very interesting points, and I do have additional questions, but I'm going to move things along a little bit more. Oh, I am so sorry. Amit, go ahead.
So I just want to come back to the enrollment in the adult trials because I think we've almost said it, but not quite said it, that one of the things that's excluded are people with active CNS lupus. And then Hermine appropriately also brought up the washout period. So are we open to allowing active CNS in the adult trials? Are we open to allowing, shorter or no washout periods? I don't think these are things that the regulatory body has imposed. I think this has been industry themselves have imposed these limitations. So I would really ask that to the industry members on the panel as to whether you would be open to relaxing the eligibility criteria in the adult studies.
I think the CNS lupus question is very related to the washout because it's a very dangerous manifestation of lupus that I think most clinicians would feel the need to treat quickly and aggressively and might lead to therefore dropout from the study or the or withdrawal of the investigational product because often it's treated with high dose pulse steroid, cyclophosphamide, B-cell depleting therapies. In terms of the washout period, I think it's very challenging to liberalize those without muddying the waters in terms of safety events. We know that there are more targeted therapies that we can know when the drug is out of your system, but that doesn't mean that the immunologic effects of that drug have completed. And I think this comes up a lot as we see more B-cell depleting therapies. We heard from a patient who struggled with kind of long-term hypogam and B-cell depletion, and we know that that does occur with some patients. So some of these things probably could be mitigated by protocol design in which you have a B-cell repopulation threshold, a level of IgG, but there's other drugs that we don't fully understand what the biomarkers are when your immune system returns to normal after that drug. So we're already in lupus drug development trying to study the impact safety-wise of another drug added on top of a conventional DMARD and steroids, which are already immunosuppressive. So I think adding the effects of past medications just gets very, very difficult to differentiate what might be a new investigational drug related and what isn't, but be hear from others as well.
I think there could also, maybe Hermine had mentioned it, some stratification by past therapies. I think, waiting long washout periods, I don't know that that works for patients and how to get them enrolled with active lupus. I think that becomes very problematic. So I think strategies like stratification could be helpful.
Returning to the neuropsychiatric lupus question, so we know that seizure and psychosis are severe manifestation of neuropsychiatric lupus, but there is like a spectrum. We did studies years ago where we saw that every child with lupus had abnormal functional MRI imaging to some degree. So there was just by virtue of the antibodies that are being present in the inflammatory milieu, the brain of a child with lupus functions differently than that of a healthy peer or their best friends. And there have a couple of studies been done. So in my mind, the question is, does the degree of neuropsychiatric involvement affect the ability or would guide the treatment? And could the treatment, that is imposed have the potential based on the mechanism of action to make that brain involvement worse?
Right, but it sounds like we need to rely on the adults and therefore change how we include or exclude CNS lupus in the adult trials if we're going to be relying even more on the adult data.
Well, if the neuropsychiatric disease would guide the adherence to the protocol, then, that would be an exclusion criteria. But I don't think that we need to be so specific.
Yeah, and I'll just add to Dr. Brunner's comment that, this goes without saying, but probably worth just saying out loud that, lupus, as we've talked about, is a systemic disease, right? Multi-organ, doesn't usually occur in isolation. And the pharmacotherapy that we use by and large also does not just affect one organ, right? If we start methotrexate, for example, for arthritis, it may also improve cutaneous disease and myositis at the same time. And so I think from a biologic pharmacology perspective, we want patients with multi-organ disease to be represented in the clinical trials. So I think the reason for excluding some of these patients, like with CNS disease, is largely from a safety perspective, right? Because putting safety of the patient enrolling in the clinical trial first. But as Dr. Brunner mentioned, there's a full spectrum, right, of CNS disease. Some is inflammatory, some's non-inflammatory. And so I don't think we should necessarily just think CNS lupus, as broad as it is necessarily a reason to exclude some of these patients from clinical trials. And so it really should be done with a safety first in mind, really thinking through the biology.
Yeah, and just to add on to those points, I do think most clinical trial protocols that exclude CNS lupus, we are talking about severe CNS lupus. I totally agree that a large proportion of patients have some degree of cognitive dysfunction, brain fog. Those usually aren't an exclusion criteria, but it's more the seizure kind of manifestations that would really drive other background therapy that often is not included in a protocol.
Thank you. I just wanted to ask maybe a clarification question. So I'm personally not opposed as someone who does a lot of drug development work to include adolescents, for example, in a phase 3 program. But I think I could foresee, and our dermatology division, for example, already allows for that often, that 12 and up can go into the dermatology trials. Brings up another issue that we're starting to parse out cutaneous lupus as well. So how is that going to play out in all of this? But that said, if we ran another pediatric trial where, let's say, we're looking at the 5 and up, I could foresee where, let's say, what does that look like? Are we still focused on extrapolation? Are we now committed to doing randomized trials? Because what if the phase 3 program fails in adults, which has happened many times before, and the pediatric program looks like it succeeded? And what are we looking at then in terms of the overall safety database, the overall proceeding with the approval of the drug? And again, it's a provocative question. I don't know that we have the answers, but I just want to bring that up as a point of discussion.
I'll just say we have thought about this question a lot also in dealing across divisions. The whole idea that, I mean, again, I've heard it over and over again the last day and a half, is there's not that much difference really between a 5-year-old and a 25-year-old that we're talking about, if we're talking about lupus, right? If we're talking about SLE. So if we choose to enroll a population ahead of time, or sorry, pre-approval, pre-original approval, where that cutoff is has to be justified with, what information we have and where we feel comfortable going. And so a lot of times, when we wrote that guideline, we said adolescents and adult trials because we know, based on the data that we've accumulated over time, that the PK between an adolescent and an adult is almost never different. Like we don't even actually do standalone PK studies in adolescents anymore because we believe that the adult data are applicable, are completely extrapolatable to the adolescent population. Where that starts to change is in childhood. And so you're gonna have to do some PK, get PK. You're gonna have to get that in the 5 to 12-year-olds or below that age, below about the age of puberty, right? So, or adult body size is really more, I think. So I think to get back to the idea of, well, where do you decide and what risk do you take? I know that from a I don't know this actually, but I assume from a financial perspective, if you're not sure your asset's going to get approved, and then you've already sunk a whole bunch of money into doing studies in children, then that increases your risk in terms of what you've lost. I don't know what that calculus is. All I'm saying from a scientific perspective and from an access perspective, we'd like as many of those patients to have that drug available upon original approval.
I have never seen that any shareholder of a drug company had to give money back to the headquarters because the company lost money. You know, the concern about excessive loss due to a small pediatric study. Now we're talking, the largest pediatric study of children with lupus was 73 patients. So how much cost is a 73 patient study compared to thousands that are enrolled in the adult program? And I think we need, in the scientific community and also under the regulator, we need to protect the children. And, you know, it's a societal task. And the share price is like, you know, a remote concern of mine.
So Lily, I see you're at the microphone, and I also saw that there were some questions online that I can't see, so I don't know if.
Yeah, actually, that's
Okay.
I was going to pose one of the questions. I think this has been addressed, but it might be interesting for the panel to opine. Would IRBs and ethics committees accept putting pediatric patients into clinical trials of a drug that up to that point has only been developed in adults through phase 2?
Prospect of direct benefit.
I think we do. And actually, quite often, what we see now is that we usurp in pediatrics medication that are available and marketed for our rare pediatric rheumatic diseases, just to get early access. So we do individual INDs to get access to the medication. Examples are some of them that treat macrophage activation syndrome. You know, there are also already off-label use of cellular therapies in children, and we do it all the time because we extrapolate the efficacy and safety to some extent from the adult data to the pediatric data.
So one other point I kind of want to bring up, and I heard it from the patient perspective yesterday, was that patients that enroll into these trials, they enroll because they're not having a good response on standard of care treatments. They're failing standard of care. And so they're looking for something that's going to help. So I think that also should be kind of thought about as the risk of, you know, some preliminary phase 2 data is that these are patients that are struggling and they're not doing well with what we have available.
Just to clarify, I think this is an important distinction, maybe the bright line. So prior to a drug's approval, right, it's not an adult approval, it's or an overall original approval. It's not available to anybody, right, except for through a clinical trial or expanded access or, you know, patient IND. So that's why there needs to be protection in that development phase for children who might want to be enrolled. And so that ethical question is, we have to, any treatment trials likely going to be more than a minor increase over minimal risk. And therefore, the standard is that you need to have evidence of prospect of direct benefit. Prospect of direct benefit does not mean that it's approved in adults. Prospect of direct benefit means that there is evidence out there that suggests that the drug would be useful in that particular patient. And usually, if you're going into phase 3, you have some data already, clinical data, not just, you know, non-clinical animal data or in vitro data. So I think that to answer that question directly, once a drug is approved, then that's practice of medicine. So it really is not an issue of, you know, that's a clinical care call.
If you look at the number of approved medication, and Stacy and Stephen, please back me up, that are approved for children with pediatric rheumatic diseases, we do a lot of off-label use. And there was a study with Dr. Schaumburg where we looked at off-label use in juvenile arthritis, and almost every child gets off-label use because we don't have that medication. As a pediatric rheumatologist, I'm delighted to have solid phase 2 results to be willing to get this medication for my patient.
Yeah, and so I think, again, we're kind of going, and I like this, we're kind of organically going through these questions. And so I think we kind of are starting to go into this part where pediatric trials have historically not been initiated until adult development is complete or after adult approval. I think we've heard some of these considerations, but are there any other scientific, ethical, operational considerations that should be thought about for, and I'm not going to say an adolescent cohort, I'm going to say a pediatric cohort in an adult clinical trial?
Ask a question with you.
Sure.
So the belimumab adult program, you got phase 3 study evaluated 2 doses, right? High and low. If the pediatric study was conducted in parallel, would we be okay with enrolling patients at 2 dose levels? How would we approach that, I guess, differently?
I think belimumab is special because there was no real dose response relationship in the company because of a, if I recall it correctly, because of a lack of a safety signal, just went for the higher dosages to go forward in the phase 3 program. So again, we look at the data, the safety and efficacy data from the adults to see whether that would be a medication we are interested in pediatrics. But given the similarities of the disease across the age spectrum, there would be no reason why we would want to have a different exposure. We talked about the different sub-analyses already.
Okay. So now I'm going to go back up because I don't think we talked about this too much. So what study designs do we think are appropriate for collecting pediatric data needed to support any extrapolation from adult data? Go ahead, Stephen.
Happy to get us started here, but just reflecting, I think, on all the conversations so far, including safety, I think what we've heard from the patient community and from the physician community is that placebo exposure is not considered, acceptable in children with lupus, and so open label designs, are definitely preferred, at least from what we've heard. And so I think as Dr. Yao mentioned, in younger children, pharmacokinetics data is still likely going to be needed. While adolescent PK is not different from adult PK, as you get lower in ages, the body size differences can be significant. And so for me, open label PK and then plus or minus PK PD, if you have a good biomarker, is really an excellent opportunity to make sure you have an optimized dose. And then, of course, safety can be collected during that same time. So as a clinician, open label PK PD safety study seems to be really walking the line of sort of optimal risk benefit. The last thing I'll sort of mention, just sort of reiterate what we've heard yesterday, is that time is damage, right? And time is damage, and damage is life. And so we can't compromise science. We have to make sure that we have, the right data to make an informed regulatory decision. But there is this need to sort of balance that with not requiring extraneous data, right? Because that time and that complexity does add cost to our patients. And I think we've heard from patients yesterday, some of whom may be in this room, that, we need therapies now, right? And each day that there's a delay in effective therapy, is palpable when we walk into a patient's room.
If no one else has anything on that topic, I kind of have it. Okay, go ahead.
One,
yeah.
Maybe, Stephen, I don't know if it's for you though, is
agree with your statement about PK/PD studies. I think still that seems like maybe questions remain from health authorities or elsewhere about whether that the efficacy from those studies is sufficient. I know that your work has shown, similarities for exposure response, but if it's always acceptable to, and obviously we've been talking about extrapolation, and based on those discussions, efficacy should be able to be extrapolated, but it seems like there may still be pushback on that. And so how to justify? Sorry, go ahead, Hermine.
Yeah, I think that's a fantastic question, and of course that's that's for our FDA and EMA colleagues to ultimately weigh in on. But again, I think the balance here is really putting the burden on the adult clinical trials to show that exposure response, across again all of these areas of uncertainty, whether it's certain organ manifestations, certain levels of disease activity. But then that same exposure response or PK/PD can really be confirmed even in an open label setting, again, especially if there's a strong biomarker for PK/PD. And so I think even with the uncertainty that I've heard, in my mind, an open label PK/PD study still seems like a good approach to address that uncertainty. But I'm curious what others think.
Stephen, you said it perfectly well. I think we already, touched on that point a couple of times. I think what we did not touch on was the perceived problems in designing a study that it can be, conducted internationally. And given the prevalence of lupus in children, an international study will be needed. And, there seem to be some, differences between the regulatory agencies in terms of the designs and also with the timing. So the timing, I think, is essential because as we saw is if we start studies in children with lupus after the medication is approved, the feasibility is really restricted. And if we don't get the medication study completed, then it will not be approved, and then the children will not have access to it in this time of, specialty pharmacy regulations.
So I guess I have a bonus
Raj, can I just make a comment?
Yes, go ahead.
This is Kalyani. So I agree with what has been said about the single arm open label trial and all that, but I think just a different perspective. I mean, to address some of the residual uncertainty that is inherent, we know in extrapolation, and also acknowledging some of these epidemiological constraints. I believe that an appropriate study design is a randomized controlled trial, right? Where we can incorporate a Bayesian dynamic borrowing framework. Now, this approach, we've heard it in different presentations, will enable us for the modulated sharing of adult efficacy data and thereby, reducing the required pediatric sample size without compromising statistical rigor or regulatory credibility. And we also know that HTA does require, comparative evidence here. So we've heard about, ethical concerns regarding pediatric exposure to a control arm. Here, what we can do is maybe, adjust the randomization ratio. Instead of using a traditional 1-to-1, use a 3-to-1 active to control ratio here. And this will ensure that you have a greater proportion of participants receiving the investigational treatment. You may have some scenarios where, the pediatric dose has not been confirmed. So here we can think of a seamless adaptive 2-part trial design, where in part A, you can have a limited cohort of pediatric patients that can be enrolled and to confirm the appropriate dose. And how do we have a starting dose? We can use the POP-PK modeling and simulation that you can derive from the adult trial data. Now, the goal of this dose selection step would be to identify a pediatric dose that achieves systemic exposures comparable to those observed in the efficacious dose. You do that, this is a scientifically sound and well-established breathing strategy. Then in part B, then you can go and proceed with a randomized trial. I mean, if we
are able to get away with just doing a PK/PD, I'm all for that. But if there are some uncertainty that going with a randomized controlled trial with limited sample size will definitely give us more robust data.
So I kind of want to get back to this point in a little bit. I have another question that I want to ask, but Lily, I'll have you ask first, and then I'll come back to this.
It's not a question, but we got several comments regarding global alignment. One was that global drug development means pediatric studies have to meet regulatory requirements in all global jurisdictions for approval. So there can only be one trial on pediatric patients worldwide, and it's not possible nor ethical to perform different studies for different regions. Then we have received 2 comments from our EMA colleagues that I think are relevant. Including adolescents in adult trials is generally accepted by the EU unless the safety is so concerning that proof of a favorable benefit risk in adults is desirable before exposing adolescents. And then the second comment is that an exposure matching approach, so just a PK safety approach, can be one of the options, absolutely, but it really cannot be a one-size-fits-all approach. So the design of studies in children and adolescents with SLE needs to be based on available data and gaps in knowledge.
Okay, thank you. So I guess one thing that I've heard over and over and over for pediatric studies is that we can't accept placebo control. I think there's been a lot of discussion on that. And so my question, I think, is kind of a mashup of a bunch of the different presentations, and I think Martine's presentation really was a great presentation. So my question is, one solution that I have not heard, and it might be because we're kind of in the infancy of getting drugs approved, is active comparator. I haven't heard anything about active comparator as a solution. And, you know, I was thinking about this specified threshold. I think the question would be, oh, well, now we have to develop a non-inferiority margin and this study's going to be monstrous. Like, but if you have this specified threshold analysis, maybe you don't have to have a huge study and you could have an active comparator. So I just want to get the panel's thoughts on using an active comparator so then you don't need a placebo.
Well, you not only, in essence, you continue standard of care when you are randomized to placebo. So
But,
what would be the active comparator?
Well, so I think that's not true because I think there are a lot of prohibited medications. So this would allow a prohibited medication to be used as the active comparator.
So almost like a natural history study, wouldn't it be? Because then you would adjust therapy as per best clinical knowledge to optimize outcomes and control disease. So that would be an option. So if I, you know, in my mind, an active comparator could only be an approved medication. And B-cell blockage may not be the right thing for every patient.
Yeah, I think it raises a lot of challenges of what that active comparator would be, given how few approved therapies there are, and the fact that children who are considering enrollment in a clinical trial typically have failed a lot of therapies and may have already failed that approved therapy, or they've been on off-label drugs that may still be active, like B-cell depletion. So it's hard to know, what you could realistically design in terms of an available active comparator that the participants haven't already been exposed to and not done well on. And I think it also raises a question, given that you have all of this background therapy, and we do now have some registry data and other data on kind of childhood onset disease outcomes. How is that different than just using existing external control data?
Yeah, I think it goes back to the question of what is the goal? Is it to show safety? Is it to show equivalent efficacy, superiority? I think it's really a question of what that arm is being used for.
I wouldn't reject active comparator out of hand. it would imply that you'd have to enroll the patients earlier, right? So they wouldn't have failed every approved drug or available drug. You would just, you'd set the bar for when those patients get enrolled earlier. So you could tell your patient that if, we've been through some drugs, we've got this other one that I could still give you, but there's a new drug that has whatever kind of data that we can show them. And then you might be able to do that. I think that would be a lot more successful than against just a standard of care. My experience recently, not in lupus, but in JIA, is that with the LIMIT trial, we ended up having to, which was PCORI funded, we had to eliminate the randomization because even the patients that agreed to be in the study, and a lot of them didn't agree because they didn't want to be randomized, but they wanted to get the drug, which is why they were willing to participate in a study. And if they didn't get randomized to it, a large percentage, I'm trying to remember right exactly now, but 25, 30%, 40% of the patients within weeks dropped out of the study.
Interesting point. So if you play it through, that would mean that you would have a patient who's, fails, let's say, the initial therapy, even not even mycophenolate is approved for children with lupus. But once it comes to the escalation to disease modifying, then you could look whether, whether you get the traditional disease modifier or the new medication. Actually thinking about your point, you could get those data from the adult trials. Now, do patients, is the response to the new medication stronger than the monotherapy studies we have for instance, mycophenolate response? Akin what has been done in rheumatoid arthritis for adalimumab and methotrexate only.
Oh, sorry, go ahead, Stephen. Please, go ahead.
I was also just going to add, I think from a clinician standpoint, head-to-head clinical trials of active comparators, can be very, very useful. But those oftentimes, I would say, should be adult studies instead of pediatric studies to at least give us that initial data. The other thing I'll say, I'll sort of answer a question by asking a question for our industry colleagues, is that I've experienced some hesitancy for some sponsors to have an active comparator with an on-label, non-patent drug, just because if there are two companies potentially competing against each other in a clinical trial, that may not be a financial wise decision. So I'm curious for our industry panel colleagues if they have any comments about that, because again, industry collaboration for some of these active comparator trials would be crucial.
Yeah, I kind of already spoke to what I see as some of the barriers to active comparators. Currently, I think the cost of the comparator drug and the implications of that trial is potentially a consideration, but I don't think it's the largest issue, to be honest. I think from a feasibility perspective, finding patients who are treatment naive to both the investigational drug and the active comparator would be extremely challenging. So while I see the appeal from the standpoint of reducing placebo exposure, I think that would be a very challenging population to try to identify and recruit just based on what we've seen so far.
Just a couple comments and a question from EME. I'll start with the comment and then go to the question. The comment is, in the EU, in the area of unmet need, we do accept comparison to placebo on top of standard of care, even if standard of care includes off-label use. I think we've talked about this, but I thought I would bring it up again since they mentioned it. And then the question from EME is, with an active control design, how would we identify the non-inferiority margin?
Yeah, I mean, I thought we would not do that. that was my take home. And like I said, I was kind of listening to Martine's talk, and I had an idea, but I'm not going to say that's the definite way you would go with this. But I don't think you could power like a traditional non-inferiority trial.
Yeah, I mean, I think too, thinking creatively, to your point about a threshold, would be, would sidestep the issue of having to identify an NI margin.
Right.
And then the other thing, so some of you in the audience will throw rotten fruit at me for suggesting this. But as I'm thinking about it, it may be a way to make it more palatable for, a pediatric trial to be run in conjunction with phase 3 or as part of phase 3. And that is, with this issue of placebo, is Lily's point of 2 dose levels. I mean, then, and we're not talking about, 1 dose level that we think doesn't work at all. That would be unethical. But if we really want to assess dose response in the adults and we want pediatric patients enrolled in that controlled trial, we could study 2 different dose levels.
Lynne, I'm going to push back. Somebody who got lupus with 14 and then participates in an adult clinical trial is now 30. They should have the same dose response than when they are under the age of 18. So I think many of those analyses could be done in the adult data, just knowing the subsets.
Sure, but that sort of presumes that you have to wait until you have those analyses, which, I'm talking about if we were going to enroll.
What makes you think that the disease in a less and under 18-year-old is different than that of an older lupus patient? And if an older, one of the larger older lupus population could benefit from a higher per kilogram dose based on organ involvement, like lupus nephritis, brain involvement, or anything like that, I think such a dose finding should be explored in the adult population and should be established there.
Fair. And my point is really to get these kids enrolled earlier, not to do it after. I don't think we need dose finding after you have an adult approval, but I'm talking about how do you enroll patients, younger patients, reasonably into a phase 3. And, fair that the higher dose may be, maybe we see differences or maybe we see exposure response, but the safety side is also important. So, to the very point, have we optimized the dose? Maybe that would be very important information to get, not just in the adult population, but also in the pediatric population.
Wouldn't that be a reason why we do a PK/PD study in children then, or in the pediatric population? And you could run it at the same time. And for sure, if there's any question or signal in the adult data, which we now, in some of the newer agents see, that we really don't know exactly what is the best medication, best dosage, then, that could be also explored. You could do escalation like it was done in the cluster study when we had autoinflammatory diseases, where you start with a lower dose, and if you have an suboptimal response, the dose was escalated. And then if you responded better, you stayed on the higher dose. And if there was no difference and or a safety event, you would down dose. So a similar strategy could be used. I saw Dr. Levy online showing very quickly, talking about placebo control, what I think a parallel design. And so if placebo is needed for whatever reason, because of the mechanism of action of the medication, which we haven't talked about, again, an early escape arm would be critical. So that kids have the option to get out of placebo if there is like a medical reason for it, if the disease doesn't respond, or even if they get worse on the medication.
Hi everyone. My name is Perpetual, and I'm just in clinical development. My question kind of brings us a little bit back to prohibited medications. So given that there are many adverse events in CSLE that may affect cumulative immunosuppression rather than a single investigative agent, such as all these slew of corticosteroids, Are there any current approaches to safety attribution that are sufficient, or should we be thinking differently about evaluating safety in pediatric lupus trials?
Well, I think you bring up a good point about how complicated it is to assess safety in these trials. I'm not sure it's so different from lupus specifically or pediatrics specifically. We'll never be able to disentangle the effects of multiple medications reliably. So I'm not sure I see a different approach unless others agree.
I think what is important in children is that we look at the long-term safety, and that maybe, if an adult with lupus is enrolled in a clinical trial at the age of 50, they're unlikely going to have more than 40 years exposure to the medication. If a child gets started on a medication at the age of 5, that's a different situation. There also may be an effect of the sequence of medications that are being used, and that sequence effect will be much more apparent, or in patients who are going to be needing those medications for a longer time.
I just wanted to make a comment on the active comparator aspects. So we learned this from the biosimilar trials, but I don't think anyone's necessarily opposed to having an active comparator, at least, again, not on the industry side, other than the fact that the practical fact of sourcing, sometimes the originator company will bottleneck the sourcing of the drug. And the other piece of it that's really important for people who are not maybe as aware in the room is that, for example, if that drug is sourced from Europe, FDA looks at it as an investigational comparator. So, and vice versa. So I think that that needs to be considered. I don't think that those are things that can't be overcome, but then it brings into question what are the value of those data, like looking across the population versus you have the US enrolled population, maybe they have a US source comparator, an EU source, for the EU, but then you're basically having the active comparator groups potentially. So I just want to bring that up as a consideration.
So I will say this though, like for biosimilars, we are really making sure that they're analytically similar, PK similar. So there might be some different considerations there than we would for a more traditional program, I guess. Amit?
So I wanted to follow up on Dr. Brunner's question and also, I guess, focus us on the last question that I'm not sure we've really addressed yet. So if we're going to more and more rely on the adult safety information to inform the pediatric approval, then how do we assess long-term safety pre or post-market, and particularly post-market?
You stole my last question.
Well, clearly this can't be accomplished within the confines of a clinical trial, which is looking at acute safety. So it requires collaboration. And as was definitely pointed out earlier today, there are a lot of ways to do that collaboration. I think, having post-marketing surveillance studies are important. Collaborating with large-scale registries where data are collected prospectively in patients with exposures can be very helpful. Linking to administrative data can be very helpful. I think it probably comes down to the unique exposure and what data source will provide the best long-term data.
So when one looks to Europe, in some country, for every biologic that needs to, the patients are registered, or also in the UK. So we do know who gets which medication. So they have national registries. Another country is Sweden. So in the absence of such a health system in the US, there are other options. It could be like a registry run by professional organizations like KERA, that would be one. There are also more and more electronic medical records that can be coalesced, like for PedsNet, where you get a rich data source for the safety and use of medication. So for instance, you could look, I think PedsNet has like 23 or 30 pediatric institutions in the US, And you could look how many patients who get like the investigational drug get admitted to the hospital as compared to those who do not. You could look how long is the persistent on the medication, which is a proxy for safety and tolerability. And you could compare that with patients who did not get that medication. And you could even adjust for the severity of the disease and could do some matching of the population. So I think nowadays we have a lot of options to achieve that, and one size will not fit all. So getting more data from different sources will increase the confidence with which we can say the medication is safe even in the long term.
If I could just follow up, a nuanced aspect of that is that the adult safety is not static. We obviously learn about new safety issues post-marketing in the adult safety that may or may not be relevant to the pediatric. So it's kind of a subset of the question of how do we determine long-term safety, especially when new adult safety issues arise.
Well, I would just add that it has to be intentional, right? I mean, I think your question is a great one and hard to concisely answer. But well, I think Dr. Mary Beth Sun's talk was really great in that it really placed a heavy emphasis on the need for these phase 4 registry and studies. But again, the data collection really has to be very, very intentional, but also it has to be long-term, right? Because, as we heard, these are rare outcomes, so you need large sample sizes. These have to be multi-site initiatives. You can't just do post-marketing safety at a few sites. And they have to be long-term, 10, 20, 30 years. And so to have an initiative like that requires a great deal of investment from the community, but also a great deal of funding as well. So these are not studied, to do this correctly is not easy. And the other thing I'll mention is, I think there's an increasing interest in using real-world data to try to answer these questions. I mean, we've heard of different databases and linkages and claims. But all this data can be very incomplete, right? If you're piecemealing, some safety data from, this registry or from this, reporting system or this EHR system. And while that can be very useful, it all has to be harmonized. So again, it has to be kind of prospectively, really deeply thought out. And then I'll just add what makes it even more difficult is the earlier comment we heard from one of the speakers. About all the concomitant medications, right? And so if we're doing post-marketing safety for 10, 20, 30 years, well, no longer are you looking at the effect of drug A, on stable background therapy, because that therapy is going to change over those 10, 20, 30 years quite substantially. And so these, to really study safety, you have to have very, very detailed longitudinal medication collection data. Which again is very difficult to do and requires, again, large multi-site initiatives that are well-funded.
But Stephen, don't you think that safety discussion is not specific to lupus? It's true for every disease or for every patient. So there will never be the complete follow-up of anybody. Maybe we don't even want that. Persons may not want to be tracked all the time, so we need to have a certain level of confidence, and we need to see how common a certain safety signal is, and we need to be intent to proactively plan on that. And it's not only a regulatory, it's also the academic community that we capture those data. For instance, in the MedWatch report, one easy fix could be, did you have onset of your disease during childhood or not? Because then you will get this additional information.
Sure.
So operationally, I'm looking at maybe my MedPeds colleagues, and maybe Kara too. How would you, if you could, construct a registry that crosses from the pediatric to the adult side? Because I think that's ultimately what you're going to have to do if you really want to collect, if you're serious about collecting long-term outcomes.
So it's hard, particularly in this country where we're not able to track individuals across the lifespan, but it can be done intentionally. I can give the example of Kara, where we have a database of 16,000 children with rheumatic diseases, and also have engaged a long-term follow-up program which recognizes that we won't be able to be seeing these patients in a clinical setting. We can't continue to see them that long, and also we just can't, it's not feasible to gain all of that adult data directly. So it transitions into more of a patient-reported program with the opportunity to obtain records for validation where patients are contacted. Now that there are challenges with that kind of long-term follow-up and finding ways to get robust response rates and retention, but they can be done, and they've been done successfully in different disease states. I think that's an excellent strategy.
We've done something similar for another medication, and it's hard to kind of span the pediatric adult realm. Patients are moving. So that's where I said maybe we need to look at different complementary sources. I do think that if we have the complete follow-up during the pediatric age range, that is very valuable. And then in the longer term, we need to think about different strategies. And it may be beyond the ability of the less than 400 pediatric rheumatologists to manage that for all patients with lupus. So looking at medical record data, administrative data in their entirety may be one solution.
I might have another question on this. It seemed like a lot of the long-term side effect interest was on specific things like growth and fertility, brain development, like bone health. How do we capture this in a registry setting?
Well, I think
thankfully, the outcome measures between pediatrics and adults are the same, especially now for the updated damage measures. And over there, premature ovarian failure and growth retardation will be captured. So you can actually get those data even from the adult registries.
Yeah, I would add that, there are challenges to accessing those long-term. There are some well-validated, as we all know, sort of patient-reported damage measures. They have their challenges as well in terms of maybe incomplete data or the, maybe the attribution may be different than what we expect, but there are validated measures, better than nothing, right? Where we don't have primary data to be able to make those assessments.
Lynn?
I was just going to say for things like bone health and metabolic risk and growth, it is very challenging to attribute any of those long-term effects to an investigational drug, though. So I think I would be cautious about including those as specific post-marketing requirements for an individual drug, given the data that Dr. Sadhu and others have shown about the known risks of cyclophosphamide and steroids. Unless you're really getting detailed background therapy medication, I think that's so challenging. We also know that a lot of patients with more severe refractory disease do go through a lot of on and off-label medications over the years. So I think you'd really need to think carefully about not just the setup and the data collection, but the analytic techniques and the goals of those specific studies to follow up those kind of long-term risks with any kind of reasonable expectation of attribution.
Yeah, and having an adequate understanding of the risks of the disease itself and the natural history, more robust data for that.
So reduced bone health mostly comes from steroid exposure or ongoing inflammation. So that's not unique necessarily to a single drug unless the mechanism of action suggests that it has an effect on bone, and that should be based on preclinical data and could be supported by adult data.
So I'm going to float maybe an idea, and it's a very nebulous idea in my head, but it just came to me. So, when you said, Hermine, that, there are 400 rheumatologists in the country, and that's probably a stretch for this small group. And I don't think it is the responsibility of just the pediatric rheumatology community. It's really the responsibility of the adult rheumatologists that accept patients, as they age into adulthood. And my experience as a pediatric nephrologist is that we take care of them and we don't want to give them up. And then when we finally do give them up, I'll get a call back from one of my patients who's become an adult, and they say, they told me they didn't want to see me because I'm too healthy. Because, they're talking about chronic kidney disease and heart disease and hypertension and all of that. And these kids primarily just have chronic kidney disease. So I do wonder, though, if the patient community, if that would be a place where we could really get some help from you to insist that, you know, when you transition to adult rheumatology or when you transition to an adult provider, that you tell them that there's this registry that we want to continue to be involved in. And by the way, if you have other patients that are in my situation, we really want you to consider getting all those patients in these registries. I mean, just to help this community get the long-term data that we think we need.
While you were talking, I was thinking, wouldn't it be nice if there was like some ICD-10 subcode that would indicate that there was early onset disease, whatever chronic disease it is, because I dare to say by the time we, you know, for several diseases, by the time the patient gets transferred care to adult rheumatology, sometimes the diagnosis morphs to the adult disease, and that also makes it easier to kind of do the prior authorization and so on. So if there was some code, that would be helpful.
And that's how the EHRs miss.
I think we need to take the discussion off label.
Margie is.
Just to make this discussion even more complicated, if I were the mother of a child with SLE, my main concern would be their brain development. And we've talked very little about CNS effects. As in epidemiology, the idea of, I know, measuring CNS effects is very difficult, other than with seizures. I don't know how we would measure it, never mind safety effects related to CNF. So just if you have any ideas there, I'd be curious to hear them.
I think there's a substantial need for research funding to address that. Could be part of the BRAIN initiative with more focus on inflammatory pediatric diseases.
I think I'm out of questions, guys. But I really appreciate the panel and I appreciate all the discussion here. I think this was an excellent discussion. So I think what I'll do is I'll move to closing remarks. I don't know if you guys want us, I'm fine with you guys here, but whatever you guys want to do, if you're comfortable. Oh, yes. So can we, I can't see, unfortunately. I see a little, yes, go ahead.
No, I think I've read the majority of them. The last one that came through from EMA is, in the EU, we almost always strongly recommend product registries that allow assessment of long-term efficacy and safety, giving recommendations on what variables should be collected and under what conditions. For example, disease status, severity, adverse events, co-medications, and other confounders. We also detail these issues in the EMA guideline on registry-based studies, almost always for rare diseases and new products with a new mechanism of action with anticipated concerning safety issues. And then one other, this is the only other EMA comment, and this was related to the point that Lynn made related to the 2 dose levels. In some cases, adult data may support a range of effective exposures rather than a single point estimate. This will depend on the specific substance and its pharmacological profile. Potential alternatives like dose titration approaches and trials evaluating 2 dose levels can be considered as possible alternatives in some cases in Roberto's view.
So Emily said she wanted to do the closing remarks. All right. So first off, I just want to say how thankful and grateful I am for all the support we had for this workshop and, you know, all the participants that have been here. I've seen up to 200 participants online in addition to who we have here in the room. So I'm really grateful for all the people that came together to really support this workshop. So there was a lot of planning that went into this workshop, and this was multiple different people across different stakeholders that really work together to really make this workshop as smooth as it was. So, great thank you to all of our planners, our speakers, everyone that participated. So I want to kind of go with what it seems everyone agrees upon across the stakeholders. And to me, I think it is a reason why this workshop was started was because, in pediatric SLE, there are very few drugs that are approved. And so there are limited approved options. And on top of that, getting drugs approved is not happening fast enough. It's a very slow, tedious process. And then, what I heard from the patients that spoke is there's other problems on top of that, that when you don't have a drug approved, that there's limitation to obtaining these medications. So this compounds the issue of getting needed treatments to our patients. I also heard that it's not good enough to halt disease activity. There still continue to be issues with social academic concerns, lupus fog. These are all areas where we'll probably need more research and more help as far as getting drugs developed so that we'll have a broader armamentarium for treating lupus disease in pediatrics. Then I also heard that there's difficulties in the way that studies are designed for families. So families have to make a bunch of sacrifices to participate in a trial. So it goes beyond study design. There's a lot of barriers to even being able to participate in a study. And so I do want to appreciate that we had so many rheumatologists that treat both adults and peds, and I think that was very helpful to hear the perspective. And what I did hear is that the circles of pediatric lupus and adult lupus, they completely overlap. So that was a very interesting perspective for me to hear, and I think it's important. And some of the differences that we see are kind of the amount of disease severity, but that severe disease in pediatrics is the same as severe disease in adults. And then there are some other considerations in developing children, such as brain development, fertility, bone health. These are some other considerations and sometimes side effects of medications that are currently approved or used affect some of these areas as well. And it's hard to capture some of the long-term side effects of medications. So I think some of the areas where we need to think about our adult information that we're currently collecting and how to better collect that information so that it's more applicable to the pediatric lupus population and to better inform how these drugs can be used in pediatrics. So I think that was one of my major take-homes. And I think there were a lot of important discussions and important questions that were asked, and we had some great answers. And again, I just want to thank everyone that was involved. I'm probably going to miss people, but we do have M-CERSI, And Dana Hamill, DPMH, and their project managers were excellent. They really helped us keep this moving. All of the workshop planning committee speakers and panelists, they were so engaged. I mean, I think we had a lot of robust discussion before we even started. Our EMA participants, and I'm just very appreciative for everyone's work on this, and so I want to just say thank you. We do have some certificates to hand out. Okay. So we want to bring Mia Andrade. McKenna Bells. Nia Cooper. Okay. Elizabeth Santa Cruz. And Chloe Raymond. Okay. So I think with that, we can conclude, and I'm wishing everybody safe travels back, and thanks again.